All righty, let's go ahead and get started. Any questions before we get rolling this morning? All righty, then let's go ahead and get started with our game plan. Same as it ever was. We were going to do muscle physiology in lecture, and we will do our origins, insertions, and actions in lab. We've had a long week with 3 long days, so we'll try to keep it a little bit shorter today, but we still have a lot to cover before the exam next week. 3 more assignments that are going to be due. Your PhysioX exercise 2, again, not hard, but time consuming because there's 7 activities. That means 7 lab reports. So make sure you're working on that. Remember, as I've also mentioned, this is your big weekend to study for the exam. So really you should start getting ahead on the material at this point so that you're prepared for next Thursday. Your Unit 11 review is due next week on Tuesday, as is your muscle activity handout, which kind of is basically doing the same types of things that we've been doing here in lab. I've given you some activities, and you tell me what muscles actions specifically and appropriately would be used for that. And then I've given you some muscles, and just like we're doing in lab, you have to come up with an activity that's going to help you to remember it. I remember the one thing that I would say is try to incorporate all of the actions of the muscle into it. The mnemonic only really helps you if it helps you to remember everything that that muscle does, because that's the whole point of this. All right. And then, like I said, all leading up to the lab and lecture exam on Thursday. The lab exam is a little bit longer than the previous ones. So one of the things that I've done is I have shortened the lecture exam a little bit just to make sure we have time, mostly just by taking multiple choice questions out because there's so much physiology anyway. So just wanted to give you a heads up. You will only have 2 hours and 15 minutes for the lecture exam instead of the 2:30 that you've been using. Most people haven't been using all that time anyway, but it will be a teeny bit shorter. You have a running clock, so you should know where that is and how that's going. But I did want to at least remind you of that because you're supposed to read all the things ahead of time before you get started, but I know people tend to want to dive in and not comprehend that. So I just wanted to give you a heads up warning about that as well. All right. Any questions on any of that? All right, perfect. After last class, I was thinking about what we did with the relaxation of the muscle, how we went through the whole process and saw how it would break up. It was a good reminder that this is a continuous process where it starts from start to finish. And now that we've done all the pieces, I thought that actually as a good review and study tool, we could probably do the same thing for initiation. So if our goal was to contract a muscle, all right, let's go through that whole entire process. What's the very first thing we need to do if we are going to contract a muscle? There we go. It is a voluntary act, so we are going to make a decision. What happens when we make that decision? I know it's 8. I know it's early. I know we've had a long week, but I'm doing this for your benefit. I know the process. If you guys aren't interested in reviewing it, I'm fine moving on to the new material. There we go. We're going to generate a neural action potential. And again, in the interest of time and space, I'll be abbreviating that thing, everything. Obviously, this is not how you would write it out on the exam. I want it out in sentences and spelled out and things along those lines. As we know, the neural action potential travels down the axon to the synaptic end bulb, right? What happens when that neural action potential reaches the synaptic end bulb? Excellent. The synaptic end bulb depolarizes. And the immediate effect of that, perfect, excellent. Oh, I like that, jumping ahead. Our voltage gated, we want to be careful. Yeah, there you go, you got it there. Okay, our voltage gated calcium channels And when those channels open, calcium, of course, enters into the synaptic end bulb. Excellent. What happens when that calcium enters in the synaptic end bulb? Again, if there's not interest in doing this, then we'll move on to new stuff. So there we go. Excellent. Calcium triggers, and I like the use of the word triggers because, again, remember, it doesn't actually grab the vesicle and bring it to the plasma membrane, but calcium triggers vesicles to move to the plasma membrane, and we release acetylcholine into the synaptic cleft. Via exocytosis. Excellent. That acetylcholine, does it stay there just huddled up against the wall of the synaptic end bulb? Nope. It diffuses across the synaptic end bulb. Oh, sorry, the synaptic cleft to the motor end plate. Excellent. There on the motor end plate, the acetylcholine binds to chemically gated. And again, if you want to be fancy, you can say ligand gated. Remember, both are acceptable. Cation channels. And of course, opens them. Once those cation channels are open, two things occur. What are the two things that occur? Excellent. Perfect. So the first thing that occurs is sodium enters the cell. Well, not first, but one of the things is sodium enters the cell. And the second thing that is occurring is potassium leaves the cell. However, equal amounts of both?
More of sodium.
Excellent. There is lots and lots, I think that is the technical term, of sodium that enters into the cell, and only a little potassium leaves it. This is significant because therefore the net effect is the muscle cell depolarizes at the motor end plate. All right, excellent. What happens when the muscle cell depolarizes at the motor end plate?
If it's high enough, it'll reach the threshold.
Excellent, and that is the key word, if. If is the key word, because remember, it isn't necessarily always going to happen, but if the depolarization is big enough to reach threshold, the muscle cell fires a muscle action potential at the motor end plate. And of course, once it fires that muscle action potential at the motor end plate, what happens then?
It'll travel down sarcolemma.
Excellent. Muscle action potential spreads down the sarcolemma. And not only does it spread down the sarcolemma, but what else does it spread down?
The T tube.
True, although remember, we want to start first as we're writing it out with transverse. But yeah, if you're doing an essay question, once you get transverse out once. And again, here's the other thing too, although I guess I'm not sure how appropriate this is on, it's easier to do like when you're actually writing this manually by hand with a paper and pencil. But if at, for instance, at the top of your essay question, you want to write out a key that, you know, T-tubule equals transverse tubule, MAP equals muscle action potential, NAP equals neural action potential. If you want to write out a key and then use abbreviations in the whole essay, that would be fine as well. Or just write it out once and then you can abbreviate it the rest of the time.
Quick
Either
question.
of those would be acceptable. Yes.
When we were, it's about the exam. So when we were taking the exam, I don't know if anybody else has had the issue where it shows that you can use a scratch paper per se, but if you tap on it, nothing really opens up. So I was wondering if that's something we can use or can't use on the exam.
No, you can. If you think about it, it was one of the things that I actually required you to do during the practice, just so you could see that it was there. You bring up that whiteboard, and when you bring up that whiteboard, you can make notes in there. You had to draw the blue star, right? Don't you remember
Yeah,
that was part of
but
that?
it hasn't been working. So I just wanted to check if anybody else has had that issue.
I have not been aware of a problem with that, but I can check into it.
Thank you.
Yeah, exactly. I mean, one of the reasons why I forced you to magnify the screen, one of the reasons why I forced you to pop that out and draw the star is not because I care what a star looks like, but because I want you to be aware of the tools that are there. So that's the whole point of that, is that you have those tools. If that's not working, I'll check to see if there's some known issue or something on my end. And then if not, I'll contact Proctorio before next week's exam. Thank you for letting me know. Has anyone else had a problem with that? Has anyone successfully used it during the exam? Or is nobody using it? Everybody? Nope. No, you haven't used it, or no, you didn't have a problem, Edna? Okay. All right. I will still check on it and see what the deal with is that. So, okay. So people haven't been using it, then that's fine. But I will still check it to see if there's an issue on my end on that. Thank you for letting me know. All right. So our muscle action potential is spreading down the transverse tubule. As it spreads down the transverse tubule, what is the effect of that? Well, remember, our muscle action potential is a big positive signal, so it depolarizes. Let's actually say it that way. Muscle action potential depolarizes the triad. Excellent. But there we have a fancy word. We should probably say what the triad is. What is the triad again? Well, it's the T tubule plus, there you go, the two terminal cisternae of the sarcoplasmic reticulum. Excellent. Why is it significant that we have depolarized the triad?
Is it that the terminal cisternae will reach its threshold?
Excellent. So the terminal cisternae reach threshold. And what happens when we reach threshold?
The channels will open.
Exactly. And
The
if you write that on the exam, that's great, but you're not going to get full
voltage.
credit because there you go. The voltage gated calcium channels open. Again, I know it sounds nitpicky, and I appreciate that. And again, my goal isn't to be tricky with these things. I want you guys to get used to being precise. I want you guys to get used to being specific, because this type of specificity, this type of being specific, being descriptive, is going to be very important to you as you move on with your medical careers, whatever field in medicine you guys are looking at going into. Being precise, being specific is significant, is important. So absolutely. So our voltage gated calcium channels open, and calcium rushes out of the sarcoplasmic reticulum into the cytosol. Excellent. Once that calcium gets out into the cytosol, what does it do?
Increased calcium concentration?
True. So, yep. So we have an increased calcium concentration in the cytosol. Excellent. And what's all that calcium in the cytosol going to do? There you go. Excellent. Our calcium binds to troponin because, and again, we can probably, I'm not sure this is necessarily part of the process, but we can put it in parentheses here because the sarcoplastic reticulum surrounds the myofibril. All right, so that's probably a good reminder, so that when that calcium is released, it binds to the troponin. When calcium binds to the troponin, what happens to troponin?
Changes its shape.
There you go, it changes shape. Or, like I said, if you want to be fancy, you can say it undergoes a conformational change, or it changes its shape. There we go. Excellent. When troponin changes its shape, the troponin, oops, is going to rotate the tropomyosin off, or rotates the tropomyosin, let's say it that way, rotates the tropomyosin, and that exposes the active site, or again, what we can say, the myosin binding site on the actin. Once that active site on the myosin is once that active site on the actin is exposed, what can happen now?
Is it that the myosin can grab onto the actin?
Yep, absolutely. However, again, there you go. It's got to be a primed myosin head. A primed myosin head can bind to the, oh, why is that not writing? A prime, there we go. Myosin head. You know, this is, I hate this about typing. All right, back in high school, I took typing for a week and got bored and switched to photography class, and now I've been paying for it ever since. All right, a primed myosin head binds to the actin. And again, we have a name for that process, forms a cross bridge. Excellent. After the myosin head forms the cross bridge, what happens now? Alex, I see what you write. Well, we can add, because it's a cycle, the nice thing about a continuous cycle is that once we're in that cycle, where you start in the cycle isn't quite as important. So I think we'll save the phosphorylation for a little bit later, but we will definitely add that in. Excellent. Now that it forms a cross bridge, our myosin head, and let's again call it a primed myosin head because that makes me happy to do so. Oops, too far. The primed myosin head can release its energy. And of course, it releases that energy by performing a power stroke. Of course, we need to define what that power stroke is. Excellent, and that is to pivot towards the M line. Excellent. That pivot towards the N line then pulls the actin towards the M line, which of course pulls the Z disc towards the M line. That shortens, oops, the sarcomere, which, as we talked about, then also shortens the myofibril, which of course is going to shorten the muscle cell. When we shorten the muscle cell, we are pulling on the connective tissues, and that is going to change the shape. Well, let's say it this way, generates a force, or like you said, it produces tension, and that's what moves, oops, the body through space. Excellent. And let's do all of this and set that, 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 and set that. Excellent. So all of that is doing, we are generating our force, producing our tension. Those are the things we definitely wanted to do. But one other thing occurs during that power stroke. During the power stroke, one other thing occurs. What's the other thing that occurs during the power stroke?
Is that when the prime myosin head splits into ADP and?
Nope. Although that's going to be the next step. You're absolutely right, though. So what happens is we need to make space for a new ATP so we can split it and prime again. To make space for a new ATP, we have to release the ADP and phosphate. So during the power stroke, the myosin head releases the ADP and the phosphate so that its binding site is now open. Right? So now after the power stroke, a new ATP can bind to the myosin head. And what happens when that ATP binds? Excellent. When the ATP binds to the myosin head releases the actin, right? We break the cross bridge. Now we can throw the part that you guys were talking about in a couple times. Now that it has let go of the actin, our myosin head phosphorylates the ATP, which again is just a fancy way of saying it splits the ATP into ADP, a phosphate, and energy. That splitting of the ATP primes the myosin head, and part of being primed is it pivots to point away, from the M line. Notice this whole part that we're writing right here about the myosin head phosphorylating the ATP, splitting it into ADP, energy, and phosphate, and priming, pointing away, we could have put at the top of this as well. So that could just have easily been at the top because it is a cycle, but that's the key here. We want to point out that it's a cycle. So if the regulatory proteins are out of the way, then the primed myosin head can bind to the actin, and the process continues. There you go. Just that simply, we have described the entire process of a muscle contraction. And again, notice, as I pointed out to you from the very beginning of this, it's not a hard process. None of these steps are super weird or super confusing where you stare at them for an hour and not understand them. There's just a lot of them. And so that's why, as we've talked about, we are dividing this, we're taking this long continuous process and dividing it into 3 more easily digestible pieces. What did we call piece 1 again?
Initiation of communication at the neuromuscular junction.
Right, communication at the neuromuscular junction. So that is part 1, right? So here, communication at the neuromuscular junction. Now, Where does that start in this long continuous process?
And making a decision.
Excellent. It starts with making a decision. And when does it end?
Spreads down the sarcolemma.
Yeah, we produce a muscle action potential and it spreads down the sarcolemma. So that right there is part 1. All right. What is part 2?
Excitation, contraction, coupling.
Excitation, contraction, coupling. Excellent. Where does it begin? It's not a trick question. If this is a continuous process where step 1 ends, what else happens?
Where the last one ended is where.
Yeah, step 2 begins. Exactly. So that muscle action potential spreading down the sarcolemma is where excitation contraction coupling occurs. And where does excitation contraction coupling end?
Where the active site is exposed.
There you go. Where the active site is exposed. Excellent. And then, of course, that leaves us with step 3. What is step 3?
Contractile cycle.
Contractile cycle. Excellent. Where does that contractile cycle begin?
Primed myosin head.
Well, I would, again, because it's a continuous process, I would argue that the troponin rotating off the tropomyosin, green, and exposing is also where step 3 begins. Where step 2 ends is where step 3 begins. But then you're right. Then it is a continuous process. And because it is a continuous process, it really doesn't have an end. Although, as I did point out, if we had wanted to, again, when you're talking about the description of this, if you wanted to prime the myosin head first, you wanted to prime the myosin head first, describe that part first here because it makes more sense, and then you can talk about the prime myosin head. That would have been fine anyway because, again, it's a continuous process. So either place where you would put it, if you'd put it at the top or put it at the bottom like this, it would have been fine as long as you have it in there. All right. I know this took us a little bit of time, but again, I think this is important to make sure that we understand 3 things. Obviously, we understand the material. We need to understand that and make sure we're comfortable with the material, and this is a good way to do it. We need to remember that it is a continuous process, right? This is one continuous process from when I make the decision to where I move my body through space, but we are breaking it up into 3 more easily digestible pieces. And the other advantage of taking the time to do this is, again, it emphasizes the specificity, the detail that I am looking for on these essay questions to make sure that you get full credit for this. Again, as we've talked about, you can be 100% correct in your description of things, but if you don't have that specificity, you're not going to get all the points. So you need to say not just that the neural action potential causes acetylcholine to be released, you have to tell me how. You can't even tell me that the neural action potential opens calcium channels. You need to say that it gets voltage-gated calcium channels to reach threshold and they open, right? So I want that specificity. I want that detail. That shows me that you understand and mastered the material, and that is how you maximize your points on this. Again, you guys should be sophisticated students at this point. I've been trying to make a point of emphasizing this, but yet I'm still seeing it. On some of these essay questions that are worth 8 or even 10 points, some people are just writing 1 or 2 sentences. If you have an essay question worth 2 points, worth 10 points, 2 sentences probably is not enough detail for what I need for that. So you need to really look at that and look at the points, and that should be an indication to you of just how much I'm looking for you to write. This is what I'm looking for. I'm looking for detail. I'm looking for specificity. I'm looking for those immediate steps. All right. Questions on that? All right, excellent then. With that then, Let's dive back in. And of course, we just did it with all the pretty words. And here's the pretty picture that kind of takes you through the whole thing as well. All right. So now we have beaten the dead horse. We know how to contract a muscle. As we've also indicated, and we've talked about this a little bit, what determines how long a muscle contraction is going to last for? What do you think? What determines how long you contract a muscle for? Come on, guys. Like I said, I know this has been a long week. We've covered. I think we've been here till 12:30 every day. Heck, first day because the groups, you guys were here longer than that. I appreciate that, but this is the last day, the last home stretch for the week. I will be taking it easy on you, but I need something back. All right, excellent. So one of them is ATP. Obviously, how much ATP is available is going to determine how long a muscle can contract for. What else? Amount of calcium. Excellent. That is going to be, what else? I think you guys are missing the obvious. What determines how long I contract my finger for?
How long you want it to go?
How long I want it, absolutely. I make the conscious decision. Now, let's take that a couple steps further. As long as I'm deciding to contract this muscle, I'm going to continue to produce neural action potentials. So on one hand, we can think of it in terms of the neural action potentials that are produced. How many of those that I produce, that could be one way we could think of that, how much stimulation. But the other way, if you want, that we could think about it is when we produce those neural action potentials, how does my neuron tell the muscle cell to contract? What does my neuron do to tell the muscle cell to contract? Well, it produces an electrical signal in the muscle cell, but how does it produce that electrical signal? It stimulates it at the neuromuscular junction, and what does it stimulate it with? How does the neuron actually stimulate the muscle cell?
Calcium with the charge lowering.
Well, calcium, remember, comes into the synaptic end bulb, but the synaptic end bulb doesn't release calcium to communicate with the muscle cell. It releases, there it is, acetylcholine. Right? Our muscle cell releases acetylcholine. So how much acetylcholine is in the synaptic cleft determines how many of those chemically gated cation channels open, how much sodium comes in, how long that muscle is going to be able to contract for. So these are the types of factors that influence how long a muscle is able to contract for. Oh, I didn't move it far enough out of the way. How long we stimulate the neuromuscular junction. And again, that stimulation of the neuromuscular junction comes from the conscious decision. But remember, how that really impacts the muscle cell is with how much acetylcholine is released. Or we could also think of it as how long it is in the synaptic cleft. Let's think of it this way. Right, Shark Week is just around the corner, and we have that acetylcholine esterase that's kind of like a Pac-Man, kind of like a shark that releases acetylcholine. If we were to put a massive amount of acetylcholine esterase into the synaptic cleft, would we be able to contract the muscle for very long? Right, as soon as our synaptic end bulb released acetylcholine, the acetylcholine esterase would be there to gobble it up. And if it was gobbling it up and breaking it down right away, would we be able to get a lot of acetylcholine to the muscle cell? Would we be able to stimulate the muscle cell and get it to contract for a long period of time? Come on, guys. Yes or no? No, of course not. No, absolutely not. Right. So exactly. So how much acetylcholine is released, how long it's in the synaptic cleft, those are all factors that can influence how long a muscle is able to contract for. So these are the types of factors that determine not just how long you're able to contract for, which obviously is an important concept, but also the strength of your contraction. If my goal is to pick up this pencil, right, I'm not using the maximum strength of my muscle, right? If I instead want to pick up the pad of paper, right, I can do that and have to use a little bit more, but that's going to be different than if I want to lift up the chair that I'm sitting in, right, or the coffee table that's over there, or any of the other things that we have in here. We need to be able to produce different amounts of strength as well. And again, as we've talked about, what happens is when that myosin and the actin interact, our sarcomere shortens, and that is what is generating our tension. We've talked about that, but we really haven't looked at it because remember when we talked about this, and heck, I'll cheat and leave the picture here because it'll make it easier to answer the questions correctly since we've had feel a little A&P hungover, it sounds like today, right? As we've talked about, we have these regions of the organelle that are the regions of our myofibril, things like the A band, oops, if I spell it right, the I band, the M line, the Z line, the H zone. Let's use band just for consistency. There you go. Those are the 5 of them. Am I forgetting anything? All right. Nope, I'm not. Excellent. So the question is how, or really if, the regions change when the muscle contracts. So let's figure it out. Does the A band change when our muscle contracts? Look at the difference between our relaxed sarcomere and our contracted sarcomere. Has the length or the size of the A band changed?
No.
No. Stays the same. But what about the I band? Has the I band changed? Yeah. Yes, it has, absolutely. In fact, we can be more specific. It gets shorter. Excellent. The reason it gets shorter is notice there's more overlap with the myosin. Since there's more overlap with the myosin, oops, there's less of the actin. Oops, why is that? There we go. There's less of the actin that is hanging out by itself, and so our I band gets much shorter. So then why didn't our A band get shorter? What's different about the A band? Why does the A band stay the same? Anyone figure it out? It's not a trick question. Well, what determines the A band again? Yeah, it's the A band is determined by the myosin. Right, where there is myosin, that is the A band. And has the myosin changed shape or changed length? All the myosin heads are grabbing and pulling, but has that big, thick myosin filament changed its shape? No. Our myosin filament, oops, is unchanged. And since it is unchanged, that means that our A band is going to be unchanged. All right, excellent. So our A band stays the same. Our I band got shorter. What about our H band? Did it change? Yeah. Yes, absolutely. For the exact same reason. It gets shorter because there is more overlap of actin and myosin. So that means there is less area that just has myosin by itself. In fact, it is actually possible that our sarcomere can get short enough where the actin from the two sides touch. And if the actin from the two different sides touched, what would happen to the H band? It would disappear. Exactly. Our H band can disappear. Right. Excellent. What about our M line? Is our M line changed by this process?
No.
Nope. It stays in the middle. But what about the Z lines? Are the Z lines changed by this? Yes. I wouldn't say that the Z lines get shorter, but what I would say, you are correct in that they are getting closer to each other, right? The overall size of the Z line doesn't change, but the Z lines are being pulled towards each other. Yes, they are pulled closer to each other. Excellent. So notice in our 5 regions, 2 of them, A and M, stay unchanged, whereas the other 3 regions of the organelles do change as that sarcomere contracts. And again, I know this is easy to see in an illustration like this, but it really does look like this. So if I can go ahead and clear that, that will allow us to actually, once again, notice here is that picture we've seen before. We looked at this when we first started talking about the myofibrils with our electron microscopy view, where we have an electron microscope showing the regions. Here is our M line at the center. Here is our H band. Here is our A band. Here is our I band. And here is our Z lines. This is a relaxed sarcomere. And this is what the sarcomere looks like when it is contracted. Notice the Z lines have been pulled much closer to each other, right? They were out here and here before. Actually, we can draw that. We don't usually get to do this in the classroom, but we can do it here. This is where the Z lines were before. And this is where the Z lines are now. So you can see they've been pulled towards each other, right? Notice as we go back, we have a very big H band. And now it's completely overlapped. Well, not quite completely. The picture has shown it completely, but it looks like there's still the teeniest bit of an H band here. And of course, the M line is unchanged as well as we look at that. And if we look at the edges of the myosin, right, our myosin has not changed. Those 2 edges stay the same. So our A band hasn't changed, but certainly our I band has gotten much, much smaller as well. So our I band is much smaller, but again, we'll go ahead and draw it. Notice the overall length of our myosin doesn't change. So the A band is unchanged, but the I band changes significantly. All right, questions on that? All right, now that we have understand what is happening at the individual protein level, we finally get the opportunity to start to build back up. While we've been focused at 1 myosin and 1 actin and 1 tropomyosin and 1 troponin and then 1 sarcomere, now we get to put them together. Because remember, when that muscle action potential that starts at the motor end plate spreads, it spreads over the entire length of the sarcolemma, down all the transverse tubules, stimulating all of the triads, releasing calcium over all of the myofibrils. And so all of the sarcomeres in a myofibril essentially are going to contract at the same time. So notice as that occurs, we have a myofibril that has a specific length to it. But as all of our sarcomeres get shorter, our entire myofibril gets shorter. And as our entire myofibril gets shorter, as we've talked about, that is going to produce tension. that muscle, the contracting of the, let's go back. Myofibrils contract. The myofibrils, remember, are connected to the muscle cell, so it shortens the muscle, and I just, again, want to emphasize this. It shortens the muscle cell. We are talking about the muscle cell here. Because those myofibrils connect to the muscle cell shortens. As we talked about, when the muscle cell or muscle fiber shortens, it pulls on the surrounding connective tissue and produces tension, right? It generates a force. And that force is what moves the muscle through space. Now, how much tension you can produce in an entire muscle, right? Bicep brachia, right? It's Friday, so of course that means it's arm days. So there you are at the gym doing the curls for the girls with your 175-pound dumbbells so that you can be doing those big, huge, massive curls. And how much you're able to lift is going to be determined by how many muscle fibers you have in that muscle cell, how many myosin heads you have. All of those myosin heads pulling together generates how much force you are able to generate. So in the bicep brachia, how much force you can generate is determined by how many muscle cells you can get to fire at one time. But in a single muscle, so obviously, if I just need to lift this paper, I can use 3 muscle cells that are in my arm. Whereas if I need to lift up our textbook, maybe I have to use 10. All right, so I can change how much, but I'm not able to lift up that Volkswagen Bug that our neighbor has out in the backyard because, again, I don't have enough muscle fibers in my arm to do that. I'm limited on how much I'm able to lift that way. But in a single muscle cell, can we change how much tension we produce in a single muscle cell? Or when a single muscle cell fires, it's all or nothing, maximum intensity or nothing at all? What do you guys think? Do you think we can change how much tension we produce in a single muscle cell?
I'm gonna go with you're asking, so yes.
Exactly. There you go. See, look at the sophistication. Absolutely. I'm asking the question. So absolutely the answer is yes. Absolutely. We can indeed change how much tension is produced in a single muscle cell. And there are 2 main ways that we do that. The 2 main ways that we do that is our starting point. What is the length of the muscle cell when we first start to contract it? And the second way is by how many neural action potentials we produce, how much we stimulate it. So how much we stimulate it and its starting length. Those are the 2 main factors that are going to determine how much tension a single muscle cell can produce. And so what we're going to do is we're going to talk about these 2 methods for modifying tension. Like I said, we are now up to the level. We are building back up. So now from here forward, we are now talking about effects of and control of and the movement of a single muscle cell. So we are now from no longer the protein, no longer the myofibril, no longer the sarcomer. We're now up to single muscle cells. And for that single muscle cell, we're going to talk about the length and we're going to talk about the frequency of contraction. All right, we're going to change how much tension that single muscle cell can produce. Now it is a teeny bit early, but this is a good natural stopping point. And like I said, I'm going to try not to go as long today as we've been going. So let's go ahead and take our first break here. I'm going to go get some coffee. Based on the relative sluggishness of the students, I would encourage all of you to go get some coffee as well. It is 8:48, so let's come back at 9:03. We'll take a full 15-minute break, and we will restart there. All right, see you guys in 15 minutes. All righty. Sorry, I have to always reset everything when I come back. Oops, that didn't work. Perfect. All right. So, like I mentioned, our goal now is to talk about, we're talking about muscle cells at the single muscle cell level. And we're going to be talking about the amount of tension that we're able to produce in them. And that there are 2 main factors that determine how much tension a single muscle cell can produce, how much we stimulate it, and what its overall length is at the beginning. All right. Now, again, if you think about this, Tension, if you think about it from the critical point, like we've been talking about, how much force you can put on a rope is determined by how many arms you can get on, or how many hands you can get on that rope, and how many you can pull. And so that's really the key. Tension is determined by two factors. Well, really one factor, if you think about it. It is how many myosin heads can perform a power stroke. Right? That's really the key in determining how much force a muscle cell can produce, right? Let's take a simple example. Let's look at a really basic sarcomere. There's my actin on one side. There's my actin on the other side. Here's my myosin filament in the middle. I didn't mean to do that, so get rid of that. And for simplicity, rather than actually drawing the myosin heads, I will just draw the necks of the myosin heads. Excellent. So here I have a very simple sarcomere with 4 myosin heads that can grab and pull onto actin and perform a power stroke. So they would be able to produce 4 myosin heads worth of tension, right? But what if I added 4 more? Now I have 8 myosin heads that can grab and perform power strokes. So as a result of that, I could produce twice as much tension, right? Because now instead of 4 myosin heads grabbing and pulling, 8 myosin heads can grab and pull. All right, that makes sense? Hopefully that's pretty simple and straightforward. Excellent. Now, adding myosin heads isn't necessarily something that is easy to do. But what we can control is how much our myosin and our actin overlap. Let's change the picture slightly. All right. Our myosin now has a total of 12 myosin heads. Got it? We're comfortable with that? Excellent. That one's there. That one's good. Put that there. And oops, put that there. Excellent. Now, this, let's say, for instance, is our normal starting length of this muscle. At this particular moment in time, if this is our starting length of our muscle, how many of our sarcomere, how many myosin heads can actually grab and perform power stroke? Well, this myosin filament has 12. Can all 12 grab onto actin and pull on the actin?
They're going to be doing it at a different time, right?
True, they are going to be doing it at different times. They do alternate in their doing it, but how many total, if I was trying to maximize the tension, where I wanted them all to grab at once and all pull at once, how many could actually grab and pull right now at this moment in time?
Only 8.
Only 8 of them, exactly. Just 1, 2, 3, 4, 5, 6, 7, 8. These 4 are not capable of grabbing and pulling, so those can't add to the tension. Conversely, if we stretch the muscle out, remember, we know we can stretch the muscle out without damaging it because we have that titin. So if we stretch the muscle out, How many myosin heads would be able to grab and pull now? Only 4. Notice by lengthening the muscle just a little bit, we have decreased how much tension it can produce. Conversely, if we shorten the muscle a little bit, how many myosin heads can grab and pull now? All 12 would be able to, exactly, right? So now all 12 would be able to grab and pull and perform power strokes. All right? So far, so good? So notice the length, one of the things that can be affected is how many myosin heads can bind to the actin. Obviously, the more the bind, more pressure you can produce, you can generate. More bind equals more tension. All right? That part makes sense. We comfortable with that? Less combined, less tension. But here's where things get a little tricky, and I'm going to have to cheat and change my drawing a little bit. Remember, as I mentioned, one of the things that we talked about, and we need to add one more component now that I think about it as well. That component is that myomesin that is on the midline. Remember, one of the things that we talked about is when a muscle is contracted, when a muscle is shortened, one of the things that will happen is that the, oops, no, you need to stay there. I just want that. There you go. The actin from the two sides can start to interact with the myomesin, and they can start to interact and run into each other as well. Notice at this point, at this length, if we've shortened the muscle enough, notice all 12 of our myosin heads are still going to be able to bind. However, with our sarcomere as short as this, are they all necessarily going to be able to perform successful power strokes? If the actin is interacting with the myosin and bumping into the myomesin and bumping into the actin from the other side, we're not going to necessarily be able to perform power strokes. So notice, it's not just about how many myosin heads can bind to the actin, but it's also how many can actually successfully produce power strokes. And of course, more power strokes equals more tension. So notice there are two factors that influence how much tension can be produced in relation to the length of our sarcomere. How much myosin and actin interact, and how much space there is for those myosin heads to form power strokes. Now, I've got a very simplistic drawing of this here, but your textbook actually has a really nice one. So let's go ahead and clear this and look at the pretty graph from your textbook, and we can see this. Notice here at the top, what do I want to do? I'll use my highlighter. That's always fun. Up here at the top, when we look at this pretty picture right here, notice here we have a large amount of overlap between the myosin and actin, so plenty of myosin heads can grab and pull, and there's plenty of space for power strokes. So there's lots of overlap and lots of space for power strokes. And when that's the case, that's supposed to be off, lots of space for power strokes. So when that's the case, we are able to produce the maximal intensity of tension. And notice we just have percentage. We're not actually worried about, the joules or whatever the, measurement of tension we're going to use. We're just talking about relative. This is where it is at its peak tension that it can produce. notice as we elongate the muscle, as we go this way, there is less overlap. And with less overlap, fewer myosin heads. Heads can bind, and our tension goes down. And notice we can also take this to the extreme. Notice down here, oops. In this case here, we have stretched the muscle cell out so much that the myosin and the actin don't overlap at all. And if they don't overlap at all, 0 myosin heads can grab and pull. And if 0 myosin heads can grab and pull, how much tension can you produce? None. Exactly. So notice here, our tension goes down to 0. Notice on this side, we still, as the muscle cell shortens, there's still a fair amount of room for power strokes. So the power doesn't drop significantly until we reach this point right here. Notice at this point right here, what's happening now is that now our actin is interacting with the other actin and the myomesin. And that's too big. I need to make that smaller. And the myomesin. When this occurs, now we can produce far fewer successful power strokes. And notice when that occurs, we see a rapid decrease in the tension that we can produce. And notice once again, if we make that sarcomere as short as we possibly can, then you are going to be able to produce 0 power strokes. And if you can't produce any power strokes, you produce 0 tension. Here, move that up there so it's a little easier to see. There you go. Two last things on this. Notice, as you can see from the illustration, most, and of course the keyword there is most does not equal all, but notice most muscles, oh wait, hold on, I want to use blue for that, let's use pink for that. Most of the muscles, when you are in anatomical position, most of the muscles of your body are at that optimal resting length. So they're going to be as efficient as possible when you decide to contract them. Now, of course, most is not all, right? So there are some that when you're in anatomical position are not at their optimal length. But most muscles, when you're in anatomical position, most of the muscles you have are at the optimal length to produce the maximum amount of tension. Okay? Now, I think this is a fairly easy concept to understand. And I also know that it's a concept that you're familiar with. You probably haven't thought of it in these terms, but you're probably aware of it if you've ever been at a bar on a Friday night. Because if you've ever been in a bar at a Friday night, inevitably, eventually, there is going to be some guy who's had one too many Long Island iced teas and gets a little bit rowdy and has to be removed from the place, typically after being subdued by a bouncer. And typically, when a bouncer subdues a person, they do it one of two ways. They either take their arms and pull them behind them. Notice if you take a person's arm and pull the arm behind them, you are stretching out that muscle. And when you're stressing out the muscle, you are reducing the amount of tension they're able to produce. And therefore, someone much smaller can actually stabilize and immobilize someone who's much bigger than them, because they're limiting the strength that they can generate, the force that they can generate with that muscle. Or what they do is they bear hug them. And by bear hugging them, you are shortening the muscles. By shortening the muscles, you're decreasing the amount of tension that they're able to produce. And again, you're dramatically decreasing their strength. So typically when those bouncers are going to subdue somebody, they take advantage of this concept. They're either pulling the arms behind them to stretch them out so that they're weaker, or they're bear hugging them so that their muscles are all shortened so that they're weaker that way as well. And like I said, that way you can immobilize someone who's much stronger than you by limiting their ability to produce tension. All right. Questions on this one?
I have a question.
Yes.
So what you're saying essentially, if the muscle is typically put in, Nelly, be quiet. It's put in a like sort of stress. So say like you pull your muscle and it's tight. Is that why we're not able to move it properly if we stress it at that point?
No. So what typically if you've strained or injured a muscle, so think it is similar. Okay, here's the only relation to this. You have the right idea. If you injure a muscle, typically that injury, especially at the cellular level, involves misalignment of the sarcomeres, damage to the myosin heads or the actin filaments or something along those lines. All right? That type of damage typically leads to inflammation and swelling and pain and things along those lines.
Okay.
So when it goes to, when the healthy muscle, and again, we'll just use simple, silly numbers. When your bicep brachia is happy and healthy, it has 100 myosin heads that can grab and pull. If you have injured your bicep brachia because you've broken 3 actins and you've ripped the heads off of 10 myosins or something along those lines, now only 75 myosin heads are able to successfully grab and pull. Then obviously you're producing a lot less tension with that muscle than you were able to do before when it was completely aligned and completely healthy and completely intact.
Okay. And so like if the muscle is say like too short or not relaxed, like you said, so like you don't have that much room to create.
Be careful. The one I want you to be careful with your wording. If I were to take my arm with my left arm and take my right arm and bring it up this way, all right? Notice I have shortened my bicep brachia.
Yes.
But it's still relaxed. I didn't contract my bicep brachia to be able to generate this tension. So it's here. So notice now, and again, this is one of those things that you can do at home. This is a great activity to do at home. What you can do at home is put your right arm out and have your significant other, have your little child, have whatever, put their hand on your wrist. All right? And your job is to flex your elbow. And do it at 3 points. Do it when you have it fully and even hyperextended. Try to flex your elbow as they put pressure against it. Then do it at about 45 degrees where you're trying to flex it. And then do it at 45 degrees. And you'll notice at 45 degrees, you're not able to produce as much force at that point as you were, for instance, out here at 90 degrees because you've shortened it. So it isn't that the muscle is contracted, right? As I bring this forward, I'm not contracting my bicep brachia. I've just made it shorter. And because I've made it shorter, now it's not going to be as strong.
Okay, got it. That was, yeah, that summed up my question because
okay, excellent.
Wanted to make sure I had that on the right track. Yep. Thank you.
And in fact, that's one of the things we would have actually done in the lab. You guys would have all paired off and seen how much tension you can produce at your arms at different length to find what the optimal length of your bicep break is. So feel free to do that at home with your loved ones. All right. Bear hug them, pull their arms behind them, especially if you have teenage kids like I do. All right, hog time. They love that. All right, questions on that? All right, that one's I think is pretty simple and straightforward. Now is where things get a little bit fun. Oops, and then there's the pretty pictures that go along with all of that. All right, frequency of stimulation. I am going to, well, actually, no, we're going to do this first. We have an important rule that we need to learn in this class. Now, again, one of the things we've been talking about is if that stimulus from the neuron is large enough, it produces that muscle action potential. And like I said, the advantage of this is we have the ability to modify it. We have the ability to change it. However, as a good general rule, the good general rule is that one neural action potential, one neural action potential is going to produce one muscle action potential. And that one muscle action potential produces what we call one twitch. So this is going to be a big, important rule for us to make sure we understand and make sense of. One neural action potential, oh, I don't have to put it quite so high. One neural action potential is going to produce one muscle action potential, which produces one twitch, which of course comes to the question, what is a twitch? And as we see here, a twitch is a single stimulus, contraction, and relaxation in a single muscle cell. Remember, we can't forget now, we are at the level we were talking about a single muscle cell that we are affecting and modifying and talking about here. Here are some examples of some twitches of different muscles in your body. Notice in all of these cases, what happens is we have a stimulus. When we have a stimulus, we then have a increase in tension, which is the contraction, and a decrease in tension, which is the relaxation. And we will actually go through these stages and explain what's happening and why it's happening in just a minute. But what I do want you to notice is that the lengths of these twitches are different in how long they last. In an eye muscle, and by eye muscle, I mean the muscles that move your eyes around in space, allow you to look up and down and left and right. In those eye muscles, a single stimulus produces a single twitch that only lasts about 15 milliseconds. Where's your gastrocnemius? Anyone look ahead in the material yet?
It's your calf muscle.
It's your calf muscle, absolutely. Your calf muscle is your gastrocnemius, superficial muscle, I should say, of your calf muscle. Notice when we stimulate it, a single twitch lasts about 40 milliseconds. Deep to your gastrocnemius, also in your calf region, is a large flat muscle called the soleus. And notice when we stimulate it, it produces a twitch that lasts about 100 milliseconds. So notice the length of the twitch can vary from muscle to muscle. We'll talk about why those differences occur later, but we will talk about them. But all three of them still have the same type of twitch, and more importantly, that twitch is divided into three distinct stages. All right? Let's draw a twitch and look at what this looks like. I'm going to do this on the whiteboard first. Now, I want you to take a really good look at this picture first so that as I recreate it, you know that I'm not recreating it incorrectly. All right, so let's go ahead and switch to our whiteboard. Clear that. And we are going to talk about a twitch. So we're going to draw a graph. That is one of the nice things about you guys having that whiteboard, and that might be one of the good reasons why I got to make sure that that's working properly, because drawing a twitch could be exactly the kind of thing that I might ask you to do on a lecture exam. Anytime you draw a graph, you want to make sure you label it. So over here is our tension that is being produced. And down here is our time. Can you guys see where I wrote time, or is that too low?
I can see it.
Okay, excellent. All right, I know some people are having problems seeing the bottom of the screen, so I just want to make sure. Excellent. All right, so on the bottom there is time. Excellent. Now, we won't worry about what the values of the tension are. Because again, we're not going to worry about the units, but we do need to worry about our time units. And so when we're dealing with our time units, actually, I'm going to exaggerate this because it amuses me to do so. Oops. All right, I don't have any idea what I just did there. There we go. Excellent. All right, there is our starting point. Anything a little off and unusual about our graph here?
The 0 doesn't start on the corner.
Yeah, 0 doesn't start on the corner. That is a little odd, isn't it? Notice if we went back to the picture, what we would see that actually what happens here at 0 is the stimulus. So I'm going to actually switch colors. Let's use purple for this part. And I'm going to exaggerate this way up here for here. This is the stimulus. Oh, I'm going to need to make this stuff smaller to fit on the screen. So there we go. This is the stimulus. And more specifically, this is when we generate the muscle action potential. That is actually the 0 time. When we are talking about a twitch starts, remember, one neural action potential produces one muscle action potential, and that one muscle action potential produces a twitch. So here at time 0 is when we generate our muscle action potential. But as you know, because we spent the whole week talking about it, muscle action potentials don't just spontaneously occur. First, we have to make a decision to contract it. Then we generate a neural action potential. Then that neural action potential goes down to the synaptic end bulb. Then once it gets to the synaptic end bulb, it releases acetylcholine. That acetylcholine spreads across the synaptic cleft, et cetera, et cetera, et cetera. Opens the voltage-gated sodium channels, or cation channels, allows sodium in, potassium out, but a lot of sodium, depolarizing cell, reaching threshold. If you think about it, all of this stuff that is occurring before 0 is really that communication at the neuromuscular junction. So notice it is a little weird to have the 0 offset like this, but it's just their way of reminding us that there are events that are occurring before the twitch, right? Remember our rule, and let's go ahead and put that rule back up here again because it is a good rule to remember all of the time. I'll abbreviate it this time. One neural action potential produces one muscle action potential. Oops, that's not a 1. Which produces one twitch. Excellent. That is a good rule to remember as we go through this. Oh, and I need to move this out of the way because I've not given myself enough space. Excellent. All right. So that's why our graph looks a little weird, because it is a way of reminding us that there are events that occur before the stimulus, before we generate that muscle action potential. So here at time 0, we generate our muscle action potential, and that produces our twitch. All right, we comfortable with that so far? Stunned silence means yes, I understand. Excellent, perfect. Now, as I mentioned, even though I didn't give myself a lot of room over here, but I guess we have more room to play and write on our drawing, so that'll work. Our twitch has 3 stages. The first of those stages is what is called the latent period. A good rule of thumb to remember for these 3 stages, and I'll put it here in parentheses to remind us, is these 3 stages all relate. I'm going to need to make this smaller. I think that'll help. Amount of tension being produced. So it's an easy way to remember what is happening in all 3 of these periods. And with a name like latent period, guess how much tension is being produced during the latent period? What does latent usually refer to? Delayed or, not present or something along those lines? Exactly, none. During this latent period, no tension is produced. Right? Remind me again, and let's write it down here. Actually, we'll sneak it up in the corner because, again, it helps us to remember. What actually determines how much tension is produced?
Power strokes?
Exactly. What determines it? I'm going to make this even smaller because I just want it here as a reminder. What determines how much tension is produced is the number of power strokes. Excellent. Exactly. That is what determines how much tension is produced. And that's really what we're looking at the measurement of, how many power strokes are produced. The instant our muscle cell is generating that muscle action potential, is it instantaneously producing power strokes?
No.
No, it's not, exactly. So if we were to draw our graph here, and we will use red for this. During this period, hold on. I'm going to cheat and use the straight line so that I don't have to try to draw a straight line. And again, I'm going to exaggerate the length of this to make sure to emphasize the point. These are not going to be drawn to scale based on what I've got here on the picture. But again, exaggerating it allows me to emphasize that point. During this latent period, no tension is produced. Because again, there are no power strokes being produced right away. Instead, if you think about it, when we first generate that muscle action potential, the muscle action potential has to spread down the sarcolemma. It has to go down the transverse tubules. It has to depolarize the triad. And actually, you know what? I should be putting this over here. And making it red. It depolarizes the triad, right? That opens the voltage-gated calcium channels and allows calcium out. Finally, once that calcium out, calcium can start to bind to troponin. So notice there are some events that have to occur, and if you think about what those events are, as we just mentioned, those events are basically excitation-contraction coupling. So during the latent period, excitation and contraction coupling is occurring. And again, no tension is being produced. This doesn't take long. It takes somewhere around, 1 to 3 milliseconds. So it is a relatively short period of time, but there is a brief period of time where no tension is being produced, and that is the latent period. Now, once we start releasing calcium, does all of the calcium bind to all of the troponin, moving all of the troponin, allowing all the myosin heads to all grab and all pull instantaneously at the same time, and suddenly we're at maximal tension for this muscle cell?
No.
No. But what is going to happen is first, the first one is going to bind, and then the second, and then the third, and then the 10th, and so on and so forth. So as a result of that, what we are going to see is a rapid increase in the amount of tension that we are producing. And this period of time where we get the rapid increase in the amount of tension we're producing is our second stage, or our second period of our twitch, and that is the contractile phase, or contractile period. Again, we define these periods by how much tension is being produced, and this is the period of time where we see an increase in tension. So tension is going up, and that is the key to this one. During this period of time, tension is increasing. Why is it increasing? Well, because more calcium is being released. As more calcium is being released, more regulatory proteins are moved. And as more regulatory proteins are being moved, more myosin heads can grab and pull. All right, so we get more power strokes. And of course, more power strokes mean more tension. So during this period of time, during this contractile period, we are seeing the tension inside of the muscle cell going up. Now, if we just generate one neural action potential and it just produces one muscle action potential, that muscle action potential is a big positive signal, a big, massive 100 millivolt change. But it's still just one wave. And once that one wave passes and splashes and gets everybody soaking wet, then there's nothing left. So just as quickly, this cell is going to repolarize. Now, when the cell repolarizes, as we know, we close the voltage-gated calcium channel, so no new calcium can come out. But there's still all that calcium inside the cytosol. And as we talked about, is all of that calcium instantly going to be locked away and suddenly the tension instantly goes down to zero?
It'll take time.
No, it's going to take a little bit of time, exactly. And so that period of time when the tension is decreasing is our third period. And this third period, although, hold on, I need to go all the way down to zero. So let me cheat. Try to get rid of that. Excellent. All right. So this period of time, and we should also, we'll use black to just show the division between these three areas. Cheat a little bit, give that there. Perfect. This third period of time is what is known as the relaxation period. Notice an important factor here. Remember, we identify these stages by the change in tension or how much tension is being produced. Relaxation period is not when the tension equals zero. When the tension equals zero, the cell is back at rest. What is happening during the relaxation period is there is a decrease in tension. So tension in this case is going down. So during this period, tension is going down. And so let's make that green. Is decreasing. Right. Again, let's be consistent. Less calcium. Oh, no, let's say, hold on. No new calcium is released, right? And calcium is being pumped out of the cytosol. So there is less calcium in the cytosol. With less calcium, fewer regulatory proteins are moved. With fewer regulatory proteins moved, fewer myosin can bind to actin. And with fewer myosin binding to actin, fewer power strokes are performed. And with fewer power strokes being performed, less tension is produced. And our tension goes down until, as we mentioned, ultimately tension equals zero. And once tension equals zero, when tension equals zero, the cell is at rest. Our twitch is over. So notice we have these three phases, all determined by the changes in tension. Latent period, when there's no change in tension. Contractile phase, when there's an increase in tension, and the relaxation phase, where there is a decrease in tension. Let's go back to the lecture and see the same thing, going over it again. Here, this one again, you can see drawn more to scale. So again, my numbers weren't quite as good, but we can see it. Notice here, our latent period, as I said, is only about 1 to 3 milliseconds. It is a very short period of time because if you think about it, it doesn't take very long for that muscle action potential to spread across the sarcolemma, spread down the transverse tubules, open up those voltage gate channels. It only takes a couple milliseconds. But that's what's happening. We need to spread that muscle action potential because that is the stimulus, the muscle action potential, right? This is, again, when we are dealing with a muscle in our body, we have this part before the zero period where we're making the decision and sending the signal and releasing the acetylcholine and all that. However, as you're going to be doing in your Physio X, instead, the stimulus is going to be you stimulating the muscle with an electrode. Back in ancient times, and by ancient times, I mean when I was in graduate school, we did these exercises on frogs. Our lab tech came in and dumped a bucket of frogs in the room, and the first thing you had to do is chase down and get your frog. Once you got your frog, you then had to pith your frog. If none of you have ever pithed a frog before, I will explain to it what it is. What you do is you find the frog's head and you find the frog's spinal cord, right where his spinal cord meets the head. Then once you find that spot, you take a large bore syringe and you insert that syringe into the vertebral column of that frog, basically destroying its spinal cord, making it completely paralyzed, but not killing it. And now that you have a completely paralyzed frog, you can lay it on the table, you can pin him down, you can peel the the skin off of his muscles, you can crack open his rib cage so you can still watch his beating heart, and you get to play with his cardiac muscle, and you get to play with his skeletal muscle, and you get to have all sorts of fun. And as enjoyable of an experience as that is, even if you're in the classroom, you wouldn't get to do it. So instead, we have these great lab simulators that do the same thing. So basically, you don't get to chase down your own frog, but Physio X has been nice enough to give you a little bit of a frog muscle where you are going to be stimulating it. So basically, you're starting right here by putting an electrical signal into that muscle and generating a twitch. And you're going to actually be measuring your twitches as you do that activity. And as you'll see, for the first couple milliseconds, no tension is produced. Then we have our contraction phase, where we have the increase in tension. More calcium is being released, more regulatory proteins are being moved, more myosin is grabbing onto actin, more power strokes are being performed, more tension is generated. Then we get the relaxation period, where calcium is locked up. Less calcium is available, right? The voltage gated calcium channel is closed, so no new calcium out. We locked up the calcium that's there. So now less calcium is available, fewer regulatory proteins move, fewer myosin heads can grab and pull, less power strokes are performed, and the tension decreases until we finally reach rest. And at rest, tension once again equals 0. All right. Questions on that? That is the tension. That is the twitch. End of story. I want to make a point of emphasizing this. This is the end of the story of a twitch. However, there is an important question to ask. When it comes time for me to move my body, like say, for instance, I'm picking up my coffee to take a sip of it, do I do that by producing a bunch of single twitches of my muscle that only last about 40 milliseconds, and in that way I'm somehow able to get that to my mouth and drink coffee that way? Is that how you typically move your body through space? By making a bunch of individual twitches that last a few milliseconds? No. So to move your body smoothly through space, we must use many muscle action potentials. So we must use many neural action potentials to produce many muscle action potentials to produce many twitches. So to move our space, to move our body smoothly through space, oops, this requires many neural action potentials to produce many muscle action potentials to produce many twitches. That seemed to make sense. We're comfortable with that statement?
Quick question about the relaxation phase. The
Yes.
resting, going back to the resting stage
Yep.
would be part of the relaxation period, right?
No. Once your tension reaches 0, the relaxation phase is over, and now the muscle is back at rest. So the same way contraction starts at 0 and goes up, relaxation starts it up and goes to 0.
Got it.
Yeah, once you reach 0, the twitch is over. You're back at rest. Okay? Great question. Any others? All right, so cool. We know to move our body through space, it's going to require many neural action potentials to produce many muscle action potentials to produce many twitches. But there's a problem. That problem has to do with the irritability of the muscle cell. Remind me again what it means to be irritable.
Easily interrupted.
True, that is a definition of irritable, but that wasn't the definition we used when we were talking about muscle cells. What did it mean for our muscle cell to be irritable? True, it's going to produce an electrical signal, but what did it actually mean to be irritable? Do you remember? Like you talking during grandma's stories. Grandma receives a stimulus and grandma responds. All right, that's what it means to be irritable. To be irritable means you receive a stimulus and respond to it. Right? That's what it means to be irritable. And here's the problem with muscle cells. Muscle cells, when stimulated for a very brief period of time, lose their irritability. Meaning that once stimulated, once irritated, they generate a muscle action potential, and for a very brief period of time, they cannot produce a second muscle action potential. Doesn't matter what you do with it. You hit it with a lightning bolt, you plug it into a light socket, right? Whatever you do to that muscle cell, there is a very brief period of time where it loses its irritability, and no matter how much you stimulate it, it cannot produce a second muscle action potential. Now, the good news is it only lasts about 1 to 5 milliseconds. This loss of irritability only lasts about 1 to 5 milliseconds. If you notice, if we look up at a single twitch, notice after, hold on, I want to use green for this. No, I don't want green because green's there. We'll use blue. Notice after 5 milliseconds, the muscle cell is just barely starting to produce tension. But during this very brief period of time, no matter what you do to the muscle cell, you cannot generate a second action potential. And we give a fancy name to this brief period of time where it loses its irritability. We call it the refractory period. Notice, does this refractory period affect this twitch at all? No, it does not affect this twitch at all. What it affects is our ability to produce a second twitch. Basically, it just means there's a brief period of time where we cannot produce a second twitch. So notice when we talked about the twitch, if I ask you to describe a twitch as an essay question on the exam, you do not have to talk about the refractory period, because the refractory period is not a part of this twitch. The refractory period determines when I can produce the next twitch. All right, does that make sense? This refractory period is going to be important for adding these twitches together to move our body through space, but it doesn't affect the single twitch. It affects the ability to produce a second twitch. All righty, oops, there we go, excellent. So I've made a mess over here, so I'm gonna have to clear that up, bingo. And then we can see the pretty words from this lecture. So again, refractory period is the brief period of time where the muscle loses that excitability, it loses that irritability. It cannot be stimulated again. And like I said, it lasts about 1 to 5 milliseconds. It is a very, very short period of time. All right, questions on this? Great. A twitch then determines how much tension a muscle can produce. But it turns out we can, like I said, change or even add these twitches together. There are a couple of interesting properties of muscle cells that allow us to change the characteristics of the twitch. So I'm going to draw my graph here again. Let's not use blue. And again, over here is tension. Oops. Down here is time. I am going to change the scale a little bit, though. So right here, and we'll use green for this, is that first muscle action potential. And as we know, when we first produce our first muscle action potential, we get, and I'll use purple for this, a twitch, a latent period, a contractile phase, and a relaxation phase. All right. Again, I've changed the scale, but that's the only thing I've changed. We still have our 3 parts: latent, where we have no tension, contractile phase, where we have an increase in tension, relaxation phase, where we have a decrease in tension. Now, just out of curiosity, if I were to wait 5 minutes and then stimulated this muscle cell again, so after 5 minutes of time, I stimulated it again, what would happen?
We'd go back in the refractory period?
After 5 minutes, would we still be in the refractory period?
No.
Not 5 milliseconds, 5 minutes. I wait 5 minutes, all right? 5 minutes later. Oops. I waited 5 minutes later, and then I stimulated this with a muscle action potential, just like I did the first time. So again, here's my muscle action potential.
Into the latent period?
Right, exactly. It would start into the latent period. I would get a twitch. And what do you think this second twitch would look like in comparison to the first twitch? I have a muscle cell, I twitch it, and it produces a certain amount of tension. I wait 5 minutes and I stimulate it again. What do you think happens to the amount of tension it produces? Is it going to be the same or is it going to be different? 5 minutes later, is it going to be more? It's going to be the same. If I waited 5 minutes, and again, now here we have to worry about what my drawing skills are going to do, but I think that looks close. I think that looks close to the same. it is going to be the same. 5 minutes later, we get the exact same tension as the first. So we get the same amount of tension as we did the first time. What's going on? Why is that? Hold on. There we go. Same tension as the first one if we waited 5 minutes later. No change. Again, it's the same muscle cell. We are releasing, we stimulated it and produced a muscle action potential. That muscle action potential depolarized it, open voltage gated calcium channels, calcium rushed out. Moved regulatory proteins, and myosin heads were able to grab and pull. And let's use super simple, stupid numbers, right? Let's say that this first muscle action potential caused 100 calcium to be released into the cytosol. And that moved the regulatory proteins, and that outproduced the tension that we wanted. Well, if I wait 5 minutes later and I give it the exact same stimulus, how much calcium is going to be released?
Would it be the same?
It'd be exactly the same, exactly. So what would happen is it would cause 100, calcium to be released, moving the exact same number of regulatory proteins, meaning that we get the exact same tension. And if I waited 10 minutes and stimulated again, guess how many calcium would be released? Nope, still 100. If I waited 10 minutes, yeah, okay, perfect. And what would the size of the twitch be? It'd be the same, exactly. So notice, stimulate a muscle, I get a twitch. I wait 5 minutes, I do it, I get the exact same twitch. All right? Because every time we stimulate it, one neural action potential produces one muscle action potential. One muscle action potential releases a set amount of calcium, meaning we get a set size twitch. And it's always going to be the same. Okay? That was supposed to be the easy part. All right, let's hopefully it gets better from here. Now, let's ask an easy question. Do I actually have to wait 5 minutes before I twitch it again, before I stimulate it again? No. No. So what I'm going to do here, and I want to write this out to make sure we understand what is happening. I am going to stimulate the muscle a second time. However, with this second stimulus, I'm going to do it after, well, let's say it this way, be more specific. Immediately after the first twitch ends. So if the first twitch ends, what is the tension in our muscle cell? If the twitch has ended, what is the tension in the muscle cell once it reaches rest?
Back to 0.
0, exactly. So notice the muscle cell's tension is 0. So what happens here, and let's grab my drawing, and now that the tension is 0, notice the tension is 0 again. So I'm not adding any tension to what is already there. I stimulate it, and again, I generate a muscle action potential. And that one muscle action potential is going to produce one twitch. And what's interesting is this time, we still get a latent period. We still get a contraction period. We still get a relaxation period. But notice the twitch is a little bit larger. So again, the muscle tension is 0. We're not adding tension to what's already there. We're not adding tensions together. We are twitching it, stimulating it immediately after it reaches 0, but we still get a bigger twitch with more tension. And the question becomes, why? After all, as we know, that one new muscle action potential, because we're using simple, stupid numbers, is going to release 100 calcium. And if it releases 100 calcium, doesn't that move a set amount of regulatory proteins? And doesn't that also then mean a set amount of myosin heads are going to grab and pull? So it should be a set tension. If we're still only releasing 100 calcium, why am I getting a bigger twitch? I think we have actually mentioned this once in the class before. See if anybody can put the pieces together. Well, remind me again, let's walk through it. Remind me again what happens during the contractile phase of a twitch.
The tension will go increase
Why does tension increase?
because we started with the excitation coupling process, right?
Yep. But what is it that actually causes the tension to increase? Alexa's got it.
Yeah.
Yeah, exactly. Notice this is when we release the calcium. As we release more and more calcium, more regulatory proteins move, more power strokes, and we get an increase in tension. So we are releasing calcium. Again, we're going to release a total of 100. But do all 100 get instantly released all at once? No, first it's going to be 5, and then 10 more, and then 15 more, and then 20 more until we get to 100. It's the same thing with the relaxation phase. In the relaxation phase, right, this is when the tension is decreasing because we are putting calcium away. Do we put all the calcium away instantaneously?
No.
No. Now, one more question. When tension equals 0, do you think all of the calcium has been locked back up in the sarcoplasmic reticulum?
No.
It turns out no. Even though the tension equals 0, there is still some residual calcium left. Not enough to produce tension by itself, but there is still a little bit of residual calcium left. And again, these are not the real numbers. I'm using these numbers because they're simple and stupid and easy. But let's say for argument's sake, 90 have been put away, so there are still 10 left. Okay? Notice if we then twitch it right away, stimulate it right away, we get the 100 that is released plus the 10 that's left over. So now we actually have 110 calcium inside this muscle cell, meaning we can move more regulatory proteins, meaning we can get more power strokes, meaning we can get more tension. Notice if we wait 5 minutes, by 5 minutes after the first twitch, is all the calcium going to be locked away? Yeah. So that's why at 5 minutes, when we produce a twitch, it's the exact same size as it was before. But, and again, the key to this is that we have to stimulate it immediately after the twitch ends, immediately when tension equals 0. If we do that, then what ends up happening is we have residual calcium in the muscle cell that causes a larger twitch to occur. That residual calcium means that when the muscle contracts, that residual calcium means there's going to be more calcium in the cell, more myosin heads are going to be able to grab and pull because more regulatory proteins are being moved. And it happens again. If we wait for it twitch to hit 0 and we stimulate it a third time, again, remember, we're waiting till it equals 0. We're not adding, right? We are not adding tension. We wait till it reaches 0. And once it reaches 0, we stimulate it again. And again, we'll use stupid numbers. Once again, we know 90 calcium get put away. And if 90 calcium get put away and there was 110 there, there's now going to be 20 calcium left. Plus, when we stimulate it, we release 100 calcium. So now there's 120 calcium left in the cell. And what happens to the size of our twitch?
Increases.
It gets bigger. This concept of how residual calcium can increase the amount of tension that is being produced after a muscle is twitched is this property of TREPI. Now, you probably have never thought of TREPI before, but I guarantee 100% you are aware of it. Because I guarantee every single person here has watched an Olympics or some type of track and field event, right? If nothing else, you have marveled at Usain Bolt, right? Watched on SportsCenter or something like that on that 100-yard dash. And as you watch those racers getting ready to do that race, are they standing as still as possible so that they can reserve all of their energy and all of their glucose and all of their ATP so they have all their resources ready for that burst out of the blocks? No. What do you know about those racers as they're getting ready to race?
They're continuously moving.
They are constantly moving. They're constantly jumping around. They're constantly moving. Literally up into the second they get into the blocks, they are moving. Why are they moving? Because by moving those muscles, they're getting that calcium, that residual calcium into their muscle cell so that when they burst out of the block, they have an increased tension, they have an increased force, and they're going to be faster coming out. So this is why they're in that constant movement up to the point of where they step into the block. When they finally have to be still for the beginning of the race, that's when they'll be still. But they're not still a second before that. They're constantly moving their muscles, right? Warming them up. But what really warming them up means is getting that calcium out of the cytosol, getting that residual calcium into the muscle so that when it comes time to burst out of that block, they have as much power and as much strength as they possibly can.
Where does that turn to fatigue? Because there is such a thing as too much warming up.
True, absolutely. There is such a thing as too much warming up, absolutely. And we will talk about fatigue as well. But what we're talking about here, I mean, while they're jumping and moving and swimming, they're not, I mean, and bouncing, they're not running back and forth, 100 meters at a time. They're just moving their muscles. They're just bouncing in place, right? Because again, you're right. There is a fine line between using up all your fuel and getting that calcium available, sure. But it is an important concept. Now, is there going to be a limit of how much TREPI you can get? Is there a limit to how much residual calcium you can get into a cell?
Sure.
Yeah, absolutely. So there will be a point where you will reach a maximal TREPI. And once you reach, oh, that's horrible. There will be a point where you reach maximal TREPI. At that point, every contraction after that will all be the exact same size, where again, they'll all just go to that maximal TREPI and come back down. So there is a maximal input that you can get, because if you think about it, if you have too much calcium left in your cell, well, then you're going to still be producing tension and the muscle's still going to be contracting. So we still have to get the tension down to 0. So there is going to be a maximal amount of residual calcium we can get in the muscle cell. So there is going to be a cap on a maximal TREPI. Now, again, I've done these drawings here, but let's look at the pretty pictures from your textbook that emphasize the same thing. Again, for the property of TREPI, we are getting a second stimulus that is occurring immediately after the relaxation phase. Again, the key to this is the tension must reach 0 first, right? And that's tiny, I don't like that. We are not adding tension together, right? Make sure we fully understand that. We're not adding tension to what's already there. The tension must equal 0. Right? But even with that, we are still enhancing the second twitch. The second twitch is going to be bigger. And again, it is bigger because of that residual calcium. And there is an upper limit to the residual calcium you can have in a muscle cell. So there is a maximal tension in TREPI. At that point, every twitch after that will all be at the same height. Now notice, it can take you 30 or 50 stimulations to get up to that maximal TREPI. So again, that's why they don't just move once and then ready. They're constantly bouncing, constantly getting ready so that they're at that maximal tension of TREPI when it's time to contract. All right? With me so far?
Yes.
All right. We have, go ahead.
No, I was just saying yes.
Oh, okay, excellent. All right, excellent. All right, so one last piece we need to add to the puzzle because we're still not quite there yet, right? Because when it comes time to lift that coffee cup to my mouth, I don't do a really light twitch with my arm and then a stronger and then a stronger and then a stronger and then a stronger until finally I'm able to lift the copy through my mouth. This is still not going to be the way we normally want to move our body through space with a bunch of twitches that suddenly get stronger and stronger and stronger, right? That isn't going to be an effective way of doing it. But notice one of the points I've been emphasizing is that with this property of TREPI, we're not adding tension together. But what's wrong with adding tension together? Can't we add tension together? Can't we add twitches together? Well, it turns out the answer is yes. And that's the second important property that affects our tension here, and that is wave summation. Right? One of the beauties of this system, and let's go ahead and draw my graph again. That there. Oh, stop changing. Too many colors. All right. That there. That there. Time here, tension here. So again, I generate a muscle action potential. Way too big. And that muscle action potential generates a twitch. Excellent. No new information there. But like I said, we don't have to actually wait for the cell to reach 0 tension before we stimulate it again. Is there a limit of how soon we can stimulate it? Well, I'm asking the question, so what's the obvious answer? Yes. What did we call that? We said that somewhere within the first 5 milliseconds, our muscle cell lost the ability to be irritated.
Refractory period.
Yeah, so if we stimulate it during the refractory period, are we going to get an effect?
No.
No. Right, so there is that brief refractory period, but remember, it's only the first 5 milliseconds, right? 1 to 5 milliseconds. Remember, as we talked about, depending on the muscle, some twitches can last 100 milliseconds. So that means there's plenty of time to stimulate them a second time before the twitch ends. And that's going to be the key. The key here, oh, no, didn't want to do that one there. I want to, oh, dagnabbit. All right, well, I guess we did. All right. So here's the key. To wave summate, we must stimulate a muscle cell a second time. But there are limits. It has to be after the refractory period, because if we do it before the refractory period is ended, it won't have an effect. So it has to be after the refractory period, but it also has to be before it reaches zero tension. Because if it reaches zero tension, do we have any tension to add our second wave to? No, right? So we need to stimulate it a second time after the refractory period, but before it reaches zero tension. And if we do that, we can add the two tensions together. So what happens here is I wait till after the refractory period, and after the refractory period, I stimulate it a second time. I generate another muscle action potential. And when I generate that second muscle action potential, I'm going to produce, and I'm going to color code it differently just to emphasize the difference, a second twitch. That second twitch is going to have a latent period just like it did before, but then it's going to have a contractile phase and a relaxation phase. And we've added the two twitches together. And then before it reaches zero, but after the refractory period, I can stimulate it again with another muscle action potential. And when I do that, I can then produce yet a third twitch that has a latent period. No, want yellow. That has a latent period, a contractile phase, and a relaxation phase. And notice in this fashion, I am adding the twitches together. And as I add the twitches together, I get a bigger and bigger, a stronger and stronger contraction, producing more and more tension. So in this fashion, this time I am. This is the goal. Here the goal is, right? Our goal is to add twitches together, right? That is now our goal, is to add the twitches together. And that's what we do with wave summation. Let's take a look at the pictures from the book showing the exact same thing. Again, we give it a second stimulus before the relaxation period ends, but it has to be after the refractory period. So after the refractory period, but before the muscle reaches zero tension. And when we do that, just like I drew, here we see that wave summation. And with this wave summation, we are capable of producing tensions that are far, far greater than what you would see with just TREPI by itself. So this is how we produce the big, long contractions, powerful contractions that move our body through space. We stimulate it rapidly enough that we produce lots of muscle action potentials, that produce lots of twitches that are added together. All right, questions on that?
This would end right again when we don't stimulate the
Yeah,
muscle
as
cells.
soon as you stop sending a stimulation, no more neural action potentials means no more neural action potentials means no more twitches, and the muscle would go back to rest. Exactly. Now, is there going to be a maximal tension we can produce with wave summation? Of course there is. Absolutely. There's, right? Do you have unlimited strength? Cody might, but the rest of us are mere mortals, right? So yes, absolutely, there is going to be a maximal tension that we can produce. And what's interesting is we frequently don't reach that point. Okay, I was just seeing if you're awake over there. We typically don't reach that point. Usually the way we typically, and again, this is the way we typically, I want to emphasize this, typically physiologically use our muscle cells is we do not reach, that's a little too big. We do not reach our maximal tension in our muscle cells. Right? Again, as we've talked about, contracting a muscle can be damaging to that muscle cell. It can misalign sarcomeres, it can break proteins, it can cause problems. So we don't necessarily want to be reaching them at maximal intensity. Instead, typically what we do is we reach a point where it is near the peak tension. And notice at that point, there are minor undulations in the tension at that level. Now, I know when I think about this, this seems a little confusing to me because as I'm lifting my coffee to my mouth, do you see all of those undulations as you're trying to take that sip of that coffee while you're drinking it? No, that coffee movement is nice and slow and smooth, right? In fact, I'll take it right now. Excellent. Why is it so smooth then if all the muscles are undulating when you're using them this way?
Is it because of tetanus?
Not a bad guess. We can tetanize the muscle, but that isn't the way we normally want to use it. All right, let me ask the question to you this way instead. Right, we have a very small number of people here, but even still, with 16 of us here, how many people here would be willing to sing as part of a 200-person choir? Not even with 200 people in the choir, you wouldn't be willing to do it, Cody?
Maybe?
Maybe. Again, do you have to have a great voice if there's 200 of you in the choir?
Not necessarily.
No, because everybody kind of blends together, right? When you have a large number of them blending together, then typically you don't quite stand out. Bob, but Ariel hit the key. How many people, even in a small classroom like this, would want to give us a 2-minute solo just on Zoom? Anybody willing to do that?
To be extra credit? I'm kidding.
Oh, someone volunteered.
There you go. No, exactly. So no, by yourself, those undulations would stand out. But even if I'm lifting something as small and as light as the coffee cup, I'm using lots of muscle cells to do that. Each individual muscle cell has its undulations, but they're not all undulating at the same rate. And because they're all undulating at the same rate, it kind of smooths it out and you get a nice smooth action. But what happens if you're trying to do an activity that only uses a couple muscles, like you're trying to thread that needle? When you're trying to thread that needle, it gets a little bit more challenging because as you're trying to thread that needle, right, you're only using a couple muscle cells. And when you've got a quartet instead of 200 people singing together, right, it stands out and it is more noticeable. So we do still see that. We see that when we're doing fine movements. The other time we might see it is if we do have to try to get them all to contract at the same time. You're trying to help your friend carry that couch up to their house, right? Because you helped them to move to the apartment, not knowing they live on the 5th floor. And by the time you get to floor 3, right, you're starting to fatigue and all your muscle cells are having to fire together to try to maintain that tension. And suddenly your muscles start shaking as a result of that. There you see those undulations of what we call an unfused tetanus because they're all firing in unison. So when you're using just a couple of them to thread that needle, or when you are when you're using them maximally, you see those undulations. But typically just for lifting a coffee cup, everything is nice and smooth.
When you say that all of them are firing together, do you mean that all of them that are fulfilling that action firing at the same time, or
Yes.
every muscle firing?
No, well, again, if you're trying to hold up the couch, I'm mostly talking about your arm muscles right at that point. And those are the ones that shake as a result of that, yes. So just the ones you're using, the muscle cells that you're using. Okay? Now, does that mean that every surgeon that's out there has a shaky hand when they're doing their brain surgery on you?
How many years have they been doing it?
Yeah, it depends. Hopefully not. So is it possible to contract a muscle at its maximal tension? Yes, it is possible to do that. Obviously, where we go here. When that occurs, we get a smooth, sustained contraction from that. We call this a complete tetanus, right? So one of the things, like those surgeons, again, I've mentioned I have my PhD in neuroscience. And so one of the things that we had to do to practice to get efficient at that is we had a jar, like a mason jar, and my PI, the primary instructor, you would put a dice at the bottom of the jar, and using forceps, you had to tie suture around the die. So inside of the jar, using sutures, you had to tie a bow, tie it into a bow, the suture around the die inside of the jar. So again, you're training that muscle so you have that smooth control as you're trying to thread that needle, right? This also is the instance, again, you hear stories all the time of grandmothers lifting cars off of babies to save the baby and stuff along those lines. In extreme stress, we can contract that muscle that way. Or maybe some of the other ways you've seen it on one of those like America's Funniest Home Videos or something like that, someone reaches out and grabs that electric wire. What happens when you grab that electric wire? Well, you get an uncontrolled stimulation that goes into the muscles, and all of those muscles contract at their peak level of contraction. You get that complete tetanus. And that level of intensity, that level of tension, typically is very damaging, or potentially is very damaging for the muscle cell. And so as such, it's typically not the efficient way that we're going to want to move it through space. So this is typically not how we use it physiologically, but it is possible to train it, or in extreme stress or artificially stimulated, to get a muscle to a complete tetanus.
I have a question.
I have an answer.
So in physical therapy, we would normally do like an ice and stem and like a stem unit, and you're pretty much stimulating the muscles through like electrode, like shocks and things. You know what I'm talking about, right?
Yeah, exactly. Yeah.
Yeah. So would that be like a complete tetanus where it's smooth, sustained contractions and pulses?
Yes. So when you are stimulating it artificially like that, typically what's going to happen is you are doing it sufficiently enough where it will reach a complete tetanus at that point. Now, typically those stimulations are not lasting for a prolonged period of time. So again, it is a short, brief, complete tetanus that is being produced. But yes, those things typically produce, because again, when you're artificially stimulating it, you don't have the control that you have of the nervous system. And so typically it is going to be maximally stimulated at that point.
And so like, how would that help with the recovery then? If you're artificially stimulating the muscles around it, is it just to produce like a blood flow? Is it to produce?
Basically, well, yeah, exactly. By contracting the muscles, basically by moving the muscles, the same way that stretching and things like that, you're going to increase blood flow. You're going to increase oxygen flow to the area. You're going to decrease congestion in that area by getting the muscles to contract and move. So those are the types of things that'll be beneficial. And as we also talked about, that stimulus also helps to play a role in strengthening the bones as well, and the bone attachments to the muscles as well.
Okay, thank you for answering that.
Yep, excellent. All righty, so there you go. We have now talked about ways that we can increase our tension in a single muscle cell by stimulating it, and how we stimulate it, and how rapidly we stimulate it, and also by our starting point of our length. So with that, that is everything that I wanted to talk about today from a lecture standpoint. So what we're going to do is go ahead and take our second break, and then after our second break, we'll come back and start on our lab stuff where we are going to move on to the arm. We're not going to get through the whole arm today. My goal is to get through all the muscles that have origins and insertions. So we will start with there and then see how we are time-wise and then go from there, okay? Regardless of how far we get, again, I want you looking at this stuff this weekend. One of the things that I will say is I think one of the parts that people are most challenged with on this exam, where I see people having the most problem, is on the muscles of the forearm. The forearm can be a bit intimidating because as you look at the models, you look at the illustrations, you look at the charts, there are a lot of muscles in the forearm. But the good news is you're only responsible for 6. 3 on the posterior side, 3 on the anterior side, and they're all superficial muscles. The other big key to them, and when we get to them, we'll talk about this, but the other big key to them is if you follow the tendons, if you look at where the tendons are and where the tendons go, they tell you everything about them. So like I said, I don't think we'll get to it today, but I want you looking at that and thinking about that for this weekend. All right, let's go ahead and take our break. It's 10:35. We'll come back at 10:50 unless there's any more questions. Any more questions before we take our break? All right, we will restart at 10:50. And like I said, we'll go into lab mode. So have your red pens, your blue pens, your handouts so that we can go over that together. All right, see you in 15 minutes. All righty. Any questions before we get rolling? So, as I mentioned, we are now on to the muscles that influence and move our arm through space. So we are going to start first talking about the ones that we need to know the origins and insertions on, starting first with what is commonly probably the most misnamed muscle, and that is the biceps. Right, biceps just means two-headed. We actually have, well, you have 4. You're going to be responsible for 2 bicep muscles in the body. So really it is appropriate to be biceps brachia. It is the two-headed muscle in the brachial region of the body. As we talked about with 2 heads, remember this is where you're going to see those 2 magical words, be specific. And again, when I say to be specific, it will either be in relation to the name or to the origin of those. And again, I don't want both. I want one or the other. So as you can see here, the biceps brachia is a 2-headed muscle where we have 2 different bellies. One belly over here to the left, and the other belly over here on the right. So the 2 bellies are, oh, that orange doesn't show up well. Let's see if blue does. 2 bellies over here. Notice more proximally, it is easier to see where the delineation is. But again, these are parallel muscles with these 2 bellies side by side. So when identifying them, we need to identify them specifically. And over here, the green one is the long head of the bicep brachia. And the blue one on the other side is the short head. The way I remember it is that the long head is lateral. So over here on the lateral side, lateral, long head, whereas the short head is medial. No easy way short and medial go together, but long and lateral do, so that makes it easy to remember that way. One of the nice things about this particular picture is you get a better sense of why these have the names that they do. The long head and short head are basically called that based on their tendons, right? Oh, that just be, hold on one second. No, actually, I don't want to change this yet. We'll go through this and then we'll do it at the end. The long head has a much longer tendon than the short head does. So the short head, as you can see, its tendon comes basically straight up. Actually, I should be using red for this, I guess. And it originates right here on the coracoid process of the scapula. Whereas if you notice, the tendon of the long head comes up here in that groove between the two tubercles of the humerus, the greater and the lesser tubercle, so the intertubercular sulcus. Notice it goes up over the head of the humerus and actually attaches to that supraglenoid tubercle, that roughened bump we saw on top of the glenoid cavity on the scapula. So we'll switch to the pictures of the bones and draw those in just a minute, but it is the lengths of the tendons that give us those two distinct origins. So we have two distinct names and two distinct origins. However, they both come together into a common tendon. You don't have to say common tendon. I know it's on the handout, but it's just to remind you that often when you have multiple bellies, they come together into a common tendon. And that common tendon inserts into the radial tuberosity. So both have the same insertion and both are going to have the same actions. Notice then, and I think we talked about this once before because the question was asked, the bicep brachia is a big prominent muscle sitting superficially on top of the humerus. But notice it doesn't actually attach to the humerus. It attaches above the humerus and attaches below the humerus. Right? The advantage of that is it crosses both the shoulder and the elbow joint, so it is going to affect both the shoulder and the elbow joint. Being an anterior muscle, anterior muscles are muscles that typically flex, right? The knee is the only one that's backwards. So being an anterior muscle that crosses the shoulder, it flexes the shoulder. Being an anterior muscle that crosses the elbow, it flexes the elbow. The other key is our relationship here to the forearm. Remember, we have two bones here in the forearm, the radius and the ulna. And remember, as we talked about, when we turn the hand so that it faces backwards, pronate or supinate the arm, remember the radius moves and the ulna doesn't. Remember, it's the radius that crosses. Here they're parallel. Here the radius crosses, but the ulna doesn't move. So as we look at the arms of the, as we look at the muscles of the arms, the ones that attach to the radius have an ability to be able to pronate or supinate, whereas those that attach to the ulna can't because the ulna doesn't move. Now the bicep brachia, of course, attaches to the radius, so it is going to supinate the hand. So again, if we start with our hand backwards, it is going to supinate the forearm or supinate the hand, rotating it outward so that we can hold that bowl of soup. All right? Questions on that? All right, let's look at the picture then and draw these. So again, now that we're here for the bicep brachia, we have our two bellies. So for that, we need our red origin. So we'll use red for one. The origin of the short head is here. Oops, no, wrong button. On the coracoid process of the scapula, exactly. So there is the origin of the long head, and we'll want to write at least here, long head for that. And actually, I've kind of made a mess of that. Hold on, let me clear that and start this again. I don't know what's going on there either. Stop that. There we go. All right, excellent. Let's make it smaller. All right. So again, here on the coracoid process right here, I'm not going to color the whole thing in because I want to see some differentiation. This is the origin for the, oops, short head, sorry. Short head on the coracoid process, and the origin of the long head is up here on that supraglenoid tubercle. So that is the origin of the long head. Now, as I have mentioned to you, let me stop sharing this for a second, and then I can bring it up. You are responsible for the models and the other materials in this class. Some of those you guys have been able to see nicely on your Mastering A&P, on the Practice Anatomy Lab, and some other places like that. Oops, wrong button. Remember also, and I'll go back, CRC has this virtual anatomy lab. Remember, as you look at the virtual anatomy lab, you can see the bones. This isn't the best view of it, but obviously here clearly we can see the coracoid process right here, which would be the origin of the short head. And we can't really see it, but up here behind it, up at the top of the glenoid cavity, we can definitely see the infraglenoid tubercle. That is going to be the origin of one of the tricep brachia muscles, but we need the one up there, so we can see that there. But that's actually not what I wanted to show you. We have this where it has the bones, and you're still responsible for some of the bones on the exam. But also we have muscles. So when we look at the limbs, and we look at one of the arm pictures, and let's go through some of the arm pictures that they have here. That's the one I want. Let's see if there's a better one. Nope, not the best, but it'll still serve our purpose. What we can see here, a little to the side, is the bicep brachia. So as we look at this, let's highlight it. Again, over here is the dividing line. So this part over here is the long head, whereas this point right here is the short head. On these models, one of the things that you'll see, one of the things that makes it confusing is that for the short head, it appears to have the longer tendon. Here we see the longer tendon of the short head, where I know this isn't necessarily rotated the correct way, but you really don't see the tendon of the long head. The long head actually kind of nestles into, because remember, it goes into the joint cavity. So when you look at the model, especially when you look at it straight on, it can be a bit confusing because you see all of the short tendon and almost none of the long tendon. So the long looks short and the short looks long. But you just have to remember, this one is the shorter because you're seeing all of it. And the long one goes into the joint cavity. So that's why you don't see all of it that way. All right. So again, that can be a little bit tricky, but make sure you're using your resources, using your tools to study this material, and that will hopefully help you to be successful with that. All right. So we've done the origin, we've done the insertion, we have done the actions. Questions on any of that? All right, group 6. What are you going to give us to help us to remember all 3 actions of the bicep brachia?
Well, my activity doesn't have all 3 actions, but I just was thinking of bicep curls.
Okay. Well,
so
actually, but you're actually correct. It actually does have all 3 actions. Because think of it this way. Can you do curls if you are pronated?
No.
Well, you can,
Well,
but can you lift as much weight when you're pronated as when you're supinated?
I don't think so.
Yeah, try it. Next time you're in the gym, don't go to a gym. Next time you're playing with weights at home, when you're playing at weights at home, try to do a curl pronated and try to do a curl, supinated and try to do a curl pronated. And what you'll find is that when you are pronated, you're not as strong. You're not able to lift as much weight. And if you think about why, what ends up happening is, remember, the tendon of our bicep brachia is attaching to our radial tuberosity. If we pronate the arm, what actually happens is it kinks the tendon. And it kinks the tendon so that you're not as efficient in using your bicep brachia. That's why when it pulls the arm out. It supinates as it pulls. So if you're going to do an efficient curl, you have to be supinated to do it. So you're doing that with the elbow, you're doing that supinated. You hopefully aren't swinging your arm too much with the curl. That is the only other problem with that. And that's why I always think a good example for this is bowling, right? To hold the bowling ball, you have to be supinated. For that, you are going to swing your arm to throw the ball, but then also to put that spin on it at the end, you got to flex your elbow as well. So again, you want all of those actions in your activity. All right, perfect, excellent. All right, questions on the bicep brachia? All right, let's switch then to, again, the bicep brachia is the muscle that gets all the praise, gets all the attention because it's superficial, it's on top. As I said, it's the one you do the curls for the girls. But there is a muscle deep behind it. As I mentioned, we are able to flex our elbow. We are able to do a curl when we're in a pronated position. Not as efficiently because our bicep brachia's tendon is kinked, but conveniently enough, right underneath the bicep brachia is a deep muscle known as the brachialis. Now notice the brachialis is in the exact same location as the bicep brachia. The bicep brachia is on top, the brachialis is behind it. They're in the exact same location, but as we'll see, their origins and insertions are very, very different. So for the brachialis, what is the origin of the brachialis? Again, you guys should have the list right in front of you. These should be the easy questions. You don't get to have them right in front of you on the exam, but now you get to have them right in front of you to make it easy to answer the questions.
Front distal half of the humerus embraces insertion of the deltoid.
Yeah, absolutely. So again, a brace is the insertion. When I have things like common tendon or embraces, I will say you don't have to write that as part of your description of it. But what I am trying to help to point out to you is the relationship of these things. As we will see, our deltoid, a lateral muscle, is going to come in and basically insert right here where the brachialis begins. We'll see why that's so significant in just a minute, but it is super significant that it basically comes in. So where one muscle inserts is where the other muscle begins. And when that happens, we say they embrace, right? When two things come together, they embrace. Excellent. So there it is on the distal, anterior distal half of the diaphysis of the humerus. And where does it insert?
Coronated process of the ulna and capsule of the elbow joint.
Excellent. And again, we don't need to worry too much about the fibrous capsule of that. We just care mostly about the bone. So here we are on the coronoid process of the ulna. Notice, while the brachialis is right underneath the bicep brachia, it does not cross the shoulder, so it doesn't affect the shoulder. It also attaches to the immovable bone of the forearm. So can the brachialis pronate or supinate?
No.
No. So it doesn't cross the shoulder. It can't pronate or supinate. So how many actions does the brachialis have?
One.
Just one. Flexing the elbow.
Well, can it not hyperextend too?
No, because again, hyperextend would be an extension. So this one being an anterior muscle just brings it forward. So all it does is flex the elbow. Whatever extends the elbow has the ability to then hyperextend it. But this one can't because it flexes. It just brings it forward. All right. Again, it's only one action, but we'll come up, group one. What'd you guys come up with for this?
Hang on. I thought this was more representative of a bicep curl because
Yeah.
you're doing the flexion.
Absolutely, yeah. So this one is more that isolation of doing the curls for the girls, absolutely. And again, notice this is the one, it doesn't matter whether we are supinated or pronated, we are still able to. In fact, if we pronate, we can actually isolate the brachialis muscle to get more of a workout for that deep muscle underneath. All right, now, notice one of the things that I mentioned is you are definitely responsible for All right, done. You guys did that. You are definitely responsible for superficial muscles on our bodybuilders. So if we go back to our pretty picture here, here we see our bodybuilder, and notice very, very clearly we can see the lateral head of our bicep brachia. Very, very obvious. Same thing over here on the other side as well. It's a nice, obvious, superficial muscle. But remember what we just said about the brachialis. The brachialis embraces the deltoid. Now, this picture is a little tricky because he's got his arm tilted forward. But what we can see is that up here is his deltoid muscle. And as we mentioned, his deltoid muscle comes in and attaches right here. The deltoid is kind of this cone-shaped muscle that comes to a point. And when it comes to a point, no matter how big and how scary that deltoid gets, it is always going to point right at the brachialis. So the deltoid always points at the brachialis. Notice we can see the same thing on the other side. Here is the point of the deltoid, and it points right at the brachialis. So even though the brachialis is a deep muscle, it is pointed out by that deltoid, and so it is going to be easy to see and identify. And again, it's a little scary looking when we look at this guy, but if we go back to the pretty picture from the model, notice we see the exact same thing here in a normal size muscle. Here is that arrow-shaped deltoid, and lo and behold, because they embrace, it always points right at the brachialis. So even though it's a deep muscle, it is easy to find on the model, and it's even easy to find on our bodybuilder because the deltoid is always going to point towards it. All right? Questions on that? All right, excellent. There is a third muscle that influences and affects our elbow, and that is the brachioradialis. The brachioradialis is a lateral muscle on the lateral aspect of the arm. Here in our illustration, we see it right here. And this muscle is a little bit tricky. What's tricky about it is on the forearm. And the problem is most of the forearm muscles affect the hand and the fingers and the wrist. Of course, the key word in that sentence was most. Most is not all. So while this looks like a forearm muscle because it is on the lateral aspect of our forearm, if we actually draw the origins and the insertions of it, we can see what the brachioradialis really does. What is the origin of the brachioradialis?
Lateral supracondylar ridge of the humerus.
I know, yeah, again, I know when you guys learned all these things, it seems so stupid. Why do I have to learn this raised ridge on the side of the humerus? Why do I have to learn these stupid raised bumps above and below the glenoid cavity? I didn't pick out the ones that I thought were hardest or the ones that were meanest or randomly pull them out of the hat. I pulled the things that we were going to be using. These are the things that we are going to be using, and that's why we did it. This raised lateral ridge above the condyle on the humerus, that supracondylar ridge, the lateral supracondylar ridge, is the origin of the brachioradialis. And what is its insertion?
Just kidding. Deltoid, no, the styloid process of radius.
Excellent. That styloid process of the radius, right? You can feel it. That bump right there, follow your thumb down. That bump you feel right there is the styloid process helping to form the joint of your elbow, is the styloid process of the radius. So notice two things. This is a muscle on your forearm, And I said most muscles of the forearm affect the wrist, affect the hand, affect the fingers. But does this muscle cross the wrist? Does it go to the hand? Does it go to the fingers? No. In fact, how many joints does this actually cross?
2.
What two?
I was thinking the elbow and the forearm joint.
Well, okay, I see what you're saying there. Yes, you are right. I like that. Okay, I'll give you that. I was thinking 1 in that it crosses the elbow. But you are 100% absolutely correct in that it is on the movable bone of the forearm. And because it's on the movable bone of the forearm, you're right. It is going to play a role in either pronating or supinating the forearm. So definitely I can see where that you would think of that as a separate joint, blah, 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 a second joint. So I like that. I like your answer to that. That's perfect. Excellent. Perfect. So because it crosses the elbow, it is going to flex the elbow. It's on the movable bone, so that means it can either pronate or it can supinate the hand. Which one is it?
You can do both.
You can't do both. Can a muscle both flex and extend the elbow, or flex and extend the arm and flex instead the head?
Supinate.
No, you're actually right. It's both. I was teasing you. You're absolutely correct. It is both, right? Now, again, it doesn't make sense to be able to do both because, as I mentioned, are you going to have a muscle that can both flex and extend the elbow, or a muscle that is going to both abduct and adduct the shoulder? No. But this is the one muscle that can do both. It pronates and supinates. But that's because this is a special muscle. Remember, one of the things we keep talking about is most. And one of the things we mentioned, and I'll move my chair so that I can do this for you, is that when we are in anatomical position, most of our muscles, I mentioned, are at our optimal lengths. So it is useful to stand at anatomical position because that means most of our muscles are going to be efficient. But is this how you wait for a bus? Again, back in ancient times, you were allowed to ride the bus. And so you'd be standing at the street corner with 14 other people. And is this how you'd be standing there waiting for the bus?
Absolutely.
Well, then while you're doing that, I think people are slowly moving away from you, concerned about that. Typically, when we wait for a bus, we do it with our arm in what we call body neutral position. Body neutral position is when the thumb points anteriorly. And that's the job of the brachioradialis. You can actually experience this. What I want you to do, see if we can do this. All right, edge of the desk. You grab the edge of the desk, make a fist, put your fist underneath the desk. And as you put your fist underneath the desk and push up, you can see a muscle right on top that stands out. That muscle that stands out right on top, when you make a fist, that lateral muscle, that is that brachioradialis. It is that lateral muscle, and its job is to bring the arm into body neutral position. So notice, if you're already pronated, when you contract this muscle, it is going to supinate you to body neutral position. If you're supinated, it is going to pronate you to body neutral position. So that is why this is able to do both. It can both this would be supinate, or it can pronate because its job is to bring you to that body neutral position. All right. So yes, absolutely, it is capable of doing both. It's really the only one that can kind of do opposite actions because it's kind of a cheat. Its goal isn't to pronate or supinate. Its goal is to get the body in between, to that body neutral position. All right.
So is that why it's more comfortable to lift a weight in kind of a neutral position up and into your deltoid and then rotate when you get to the top?
Right, as you're bringing it up, because again, it allows you to better engage your brachioradialis. Yeah, the more muscles you can efficiently use in that flexion of your elbow, the more weight you're going to be able to lift. All right, group 5. What'd you guys come up with for an action to help us to remember our brachioradialis?
We came up with like shaking hands, like extending your arm to shake someone's hand.
Okay, excellent. So you're going to bend the elbow there, and you have to be in you have to have the hand in body neutral position, right? If you're the
Yeah.
queen, you can just, you know, put it out so that they kiss it. But absolutely, right? You have if you're going to put your hand out to shake someone's hand, it needs to be at that body neutral position so that you can shake. I like that. I like that one a lot.
Thing.
Right? Of course, as I mentioned, the whole. Oh, are you going to demonstrate it for us? I did, but you need to do it.
Let's see if my camera is working today.
Okay.
I can do it.
Okay.
So you just shake hands.
All right, excellent. Yeah, putting your hand out to shake. There you go. Exactly. All right. Now, again, remember, as I mentioned, right, the whole point of contracting muscles is to woo. So, right, next time, again, I know California's shutting down. You're not allowed to go to bars anymore. But the next time you go to the bar, you can do what I do, right? Back in the day, as I was trying to woo my significant other, right, I would go out there and, of course, bust out my best dance moves, right? And back in those days, of course, the most wooable of all the dance moves was, of course, the robot. And how do you do the robot? Hand in body neutral position, right? And you flex the arms. And the women just swoon when you do that, right? That body neutral position to the hand, that flexing of the elbow, and they just swoon as a result. All right. Yes. And we're not allowed to shake hands anymore. So stick with the robot. There you go. Exactly. All right. Excellent. So those are the major flexors of the elbow. Oh, and excellent, right? Again, that body neutral position. Notice our bodybuilder here is cheating a little bit on this. That's another thing that I was pointing out. You put your hand underneath the table and you make that muscle stand up. But that's also a very classic bodybuilder pose. Notice how she's turning her wrist though. So we can still, even though she's turning her wrist, we can see that there is a prominent muscle that is sticking out that lines right up with her thumb. And that prominent muscle that sticks out, that one that they make stick out when they put that pressure on the arm, is that brachioradialis, that prominent forearm muscle. The nice thing about this is not only is it easy to find, you find the thumb and you follow it straight back, but the nice thing about having an obvious, easy to find muscle is, remember, as I talked about, the forearm can be a little tricky. If you have a muscle that you're 100% confident with, then from that, you can migrate posteriorly, you can migrate anteriorly, and you can kind of find your way and be able to identify the stuff that you need to be successful that way. So it makes a good, nice, clear starting point when we get to the forearm, an anchor point to help us to figure out what the rest of those forearm muscles are. All right, questions on that? Excellent. All right. Speaking of lateral muscles, let's talk about a proximal lateral muscle, and that is the deltoid. I love that picture. I'm going to go back to it again from the model. Here on the model, it really is this C-shaped, arrow-shaped muscle that really is out here on the lateral aspect. Remember, as we talked about, this is one of those multi-pennate. I know it seems like it might want to be convergent because it all comes to the point, but this again is another place where the model does a good job of showing us. There's all these different bundles of fascicles at all these different orientations that give us this nice triangular and curved C-shaped muscle that really sits on the lateral aspect. Some of it is anterior, and some of it is posterior. And we see that in its origin. In fact, we saw another muscle that had a similar C-shape attachment, and that was the trapezius. So when we draw our deltoid, notice our origin of the deltoid is going to be the spine of the scapula, acromion of the scapula, and the lateral third of the clavicle, which, if you remember, was the exact same attachment point for the insertion of the trapezius. So the trapezius comes in and attaches at this point. And from this point, then our deltoid then continues out from this as that anterior and posterior muscle. All right. And of course, where does our deltoid insert?
Deltoid tuberosity of humerus.
Right. Remember, we saw that triangular roughened patch on the lateral aspect of our deltoid. That is where our deltoid inserts. And remember, that is also where our brachialis then attaches, again on the anterior side, but notice it embraces the brachialis on one side, it embraces the trapezius on the other. It's kind of nestled right in there in between. All right, so again, if we go back to our pretty picture, all right, so again, the trapezius would come in here, the brachialis would come down from there, and then in between is the deltoid. All right, how many joints does the deltoid cross? Just one, just the shoulder. And being a lateral muscle, what is the most obvious action of it going to be? Abduct the arm, right? Anterior muscles bring the arm anteriorly, that's a flex. Lateral muscles bring the arm laterally, that's an abduct. So absolutely, the obvious action of it is an abduction. But remember, this is a C-shaped muscle. With this C-shaped muscle, some of the fascicles are just here on the front. And if we just contract the fascicles here on the front, we can medially rotate the humerus, right? We can make our humerus rotate in medially. We can medially rotate the humerus. And if we just use the posterior fascicles, we can laterally rotate the humerus. So we can rotate it outward or we can rotate it inward. Medial rotation or lateral rotation, depending on which of the fascicles we are using. So it's going to abduct, it is going to medially rotate, and it is going to laterally rotate. Group 5, what'd you guys come up with for this one?
For the deltoid?
Yep.
We came up with like when your arms are moving, when you're walking or running.
Okay.
And then we said like as if you were going towards your delta airline.
There you go. So you're walking down the airport. I like that. I like that. I like that. I mean, you definitely do swing your arms as you're walking that way. So I think that works. Although you maybe don't abduct it that way. Again, maybe I'm biased because, like I said, I did both my graduate and my undergraduate work at UC Davis. So I lived in Davis for far more than any sane individual should have to live in Davis, California. And one of the problems with living in Davis, California, is that if you live there, and you live there for at least 6 months or more, you are actually required by law to actually spend at least an hour and a half on your bike every single day. All right, it's the truth, it's the law. And so one of the things you get very used to doing is signaling. And if you think about your bicycle signaling, to bicycle signal, you have to abduct your arm. Of course, you have the arm flexed. And then if you are going to tell people you're going to take a right-hand turn, you have to laterally rotate the humerus so that you're able to do that. And if you're going to tell people you're going to stop, you have to medially rotate the humerus so that you can tell them to stop. So that ability to medially and laterally rotate the arm while it's abducted is why everybody in Davis has huge, massive deltoid muscles. Well, really just on the left-hand side, because they're just on their bikes hours a day, just doing nothing but using their deltoid. So that's why all the Davis people look so weird. They have these massive deltoid muscles on their left side from riding those bikes and huge calves. All right. Questions on that? All right, excellent. All right. Here we have our latissimus dorsi. So chicken dance isn't bad. That definitely is going to be the abduction. I don't know how much you necessarily medially and laterally rotate for your chicken dance. I guess if you're really into it, you might do it that way, right? But I think the chicken dance is more just of an abduction. It's definitely the abduction part. And of course, my wife walks in while I'm doing my dance. Oh, excellent. Can you guys give me 1 minute? Minor household emergency. Give me 1 second. I'll be right back. All righty, excellent. Oops, there we go. Sorry about that. Sprinkler got ran over, flowing up through the air, so I had to fix that. So problem solved. All right, excellent. Let's talk about the latissimus dorsi. The latissimus dorsi is similar to the trapezius in that it is a mostly posterior muscle, right? It is superficial, like the trapezius, but it is lower, so it is inferior, whereas the trapezius was more superior in its location. As we can see when we look at the origin from the back, we can clearly see that it has a big, broad, elaborate origin. So let's switch to our bones. For our latissimus dorsi, what is the origin of the latissimus dorsi?
Thoracomular.
Go ahead.
Thoracolumnar fascia.
Okay, it is supported by that, absolutely. But what are the bony attachments?
Lower lumbar vertebrae.
Okay, so it does indeed originate from the spinous processes of the 5 lumbar vertebrae, but are those the only vertebrae that it originates from?
And the lower 6 thoracic vertebrae.
There you go. 3, 4, 5, 6. So notice, again, we could write that as T7 through L5 of our vertebrae. Excellent. Although that should probably be red. Perfect. And definitely it is there on the midline, but notice it is big and broad. There are a couple other places where some of the fascicles begin. Some of them, notice, are going to start out here on the lower ribs. Some of them are going to start over here on the crest of the ilium. Anyone else? If only I had a list of the origins in front of me.
At the crest of the sacrum and ilium.
Okay, where else?
Inferior of the scapula.
There you go. even up here on the inferior angle of the scapula. Notice what happens here. And I'll go thin lines and I'll go green. We have all these different fascicles coming from all these different locations, all coming together in forming this big, broad muscle, right? Some of them coming off the ribs, some of them coming off of the hip, some of them even coughing off the base of the shoulder pad. But, I mean the shoulder blade, but they are all coming together into a single small tendon. And this single small tendon inserts where?
Intertubercular sulcus of humerus.
That intertubercular sulcus. Remember, the intertubercular sulcus was that groove between the greater tubercle and the lesser tubercle. But if you remember, that is actually on the anterior side of the bone. So it is actually attaching to the anterior side of the bone. So while it's mostly posterior, all of these fascicles are coming together and they actually insert onto the anterior side of the bone. Notice if we look over here, we can cheat with our pectoralis major picture, because here with our pectoralis major picture, we can see the greater greater tubercle, the lesser tubercle, and remember that groove that starts there but goes all the way down the neck. And basically it is right here that the latissimus dorsi comes from the backside. All those fibers come up on the backside and come to the front of the femur. Not femur, front of the humerus. All right, so notice if we go back to the picture. Absolutely, so definitely, let's take a look at that as we look at this. So again, notice we have all of these fascicles. Notice some of them, right, coming off the scapula, some of them coming off the back, coming off the sacrum, coming off the ilium, all these fascicles together, but they go to the front. Now, you are exactly correct. Notice again, the location tells us a lot about this. This is a posterior, a medial muscle. Right? Mostly posterior medial muscle. So you're absolutely correct. Being a medial muscle, one of the obvious actions of it is to bring the arm in, that adduction. Being a posterior muscle, what is its prominent feature or prominent action being posterior? Well, if anterior muscles flex, what do posterior muscles do?
Extend.
Extend, absolutely. So being posterior, it is going to extend. Being medial, it is going to adduct. But it basically comes up and wraps around onto the front of the humerus. So when it pulls on the humerus, because it's attached to the front, notice it pulls the front of the humerus towards the midline. So because it has an anterior insertion, when it pulls on the humerus, what's the third action of the latissimus dorsi going to be?
Retract?
No, medial rotation, right? It's going to medially rotate the arm, medially rotate it, because it's attaching here, it's pulling the humerus over, medially rotating it. All right, so it extends the arm, brings it back, it adducts the arm, it brings it in, and it medially rotates it. Group 3, what'd you come up with for the latissimus dorsi?
Yeah, my partner came up with scratch your butt.
Okay. Are you going to demonstrate that for us?
Yeah, I guess so.
I have a different one if you need me to do it. I mean,
Nah, it's okay. Screw it. We're already in. Okay.
In for a penny, in for a pound.
Yeah, exactly. 1 second. I'm trying to turn on my camera. Okay, can you see me?
Yep, we gotcha.
Okay, so I guess like this. Scratch your butt.
Okay, so that definitely adducts, that definitely extends it to come back, and you have to immediately rotate it to get the finger and the thumb there. I like that. I like that a lot. That's excellent. That's hilarious. That's great. I haven't heard that one before. I like that one a lot. That's spectacular. The other thing that I think about for this, if you think about it, as you look across our species, which particular athletes tend to have the biggest latissimus dorsis? Swimmers, right? Absolutely, right? Michael Phelps practically has bat wings, right? If he holds his arm out, as you see. Because if you think about the swimming action, you are starting with the arm out here, and you're basically doing two things. You're adducting the arm, bringing it towards the midline. You're extending it, bringing it back, and you're immediately rotating to scoop the water. So if you think about it, if you do them both at the same time, you're basically doing the butterfly stroke. If you're doing them one at a time, then you're basically doing freestyle. So that ability to bring the arm in, to bring the arm back, and to immediately rotate it is that swimming stroke. So that's another one, great one as well, where you're doing that. Although I like the scratch your butt. That's easier to remember. Excellent. So that is perfect. That is great for the latissimus dorsi. All right. One more. Actually, I think we have two more. Now, notice our latissimus dorsi, remember, as we talked about, is a posterior medial muscle. Well, our pectoralis major, whoops, is an anterior medial muscle. Let's take a look at its origin and its insertion. What is the origin of the pectoralis major?
The clavicle, sternum, and the cartilage of ribs 1 through 6.
Excellent. So it starts up here on the clavicle. The sternum and the cartilage of ribs 1, 2, 3, 4, 5, 6. Anything else?
And the aponeurosis of external oblique muscles.
Right. Now, obviously, there isn't a bone for that, but we'll cheat and make it slightly pink here. Because remember, we know that aponeurosis of the external oblique comes off, and it actually inserts into that aponeurosis. It inserts into that tendon. You probably haven't thought about the pectoralis major this way, but if you think about it, this actually makes a lot of sense. If you notice, it has this very distinct C-shape origin. So as you work this muscle out, as we know, when you work a muscle out, the muscle gets bigger. Can it expand upward? Can it expand downward? Can it expand medially? No, it is pretty much locked in place. So as people get large pectoralis majors, which way do they project? Anteriorly, right? That pectoralis major stands out prominently on those bodybuilders because it can't get wider, it can't get more medially. So basically, as it gets bigger, it just comes out, becoming this big, huge, prominent muscle that sticks out. And here's where it really gets interesting. Where does it insert? That exact same intertubercular sulcus of the humerus. Notice the pectoralis major and the latissimus dorsi have the exact same insertion. So notice when you look at their actions, I'll cheat and put it over here again. We have a medial muscle here in the latissimus dorsi that was posterior, and the insertion inserts into the intertubercular sulcus. I'll just abbreviate that so that we have more room to write. As we know, because it is medial, it adducts. because it is posterior, it extends. And because it inserts into the intertubercular sulcus, it medially rotates. Well, let's look at the pectoralis major. The pectoralis major is also medial. It is anterior. And it also inserts into the intertubercular sulcus. Because it is medial, what do you think its primary action of being medial would be? Yep, you're absolutely right, Ryan.
To abduct.
Yeah, to adduct again. Bring it towards the midline. Being anterior, though, it's not going to extend the arm. Being anterior, it's going to do what? These should be knee-jerk reactions. Anterior muscles do what? Flex. Posterior muscles do what? Extend. That rule is true 99% of the time. The only thing that screws it up is the knee. The knee screws that up, but everything else, and we're not on the leg yet, everything else, anterior flexes, posterior extends. That should be a knee jerk, easy answer. So adducts, flexes, and because it also inserts into the intertubercular sulcus, it also is going to medially rotate. So notice the pectoralis major and your latissimus dorsi have two actions in common. Both adduct the arm, both medially rotate the arm, but because the pectoralis major is anterior, it flexes, and the latissimus is posterior, it extends. And you're right, there are a lot of muscles attaching, and well, these are the two big muscles, these two big muscles attach here onto the arm, which is one of the things that gives our arm a tremendous amount of strength. All right, we are down to group 5. Group 5, what'd you come up with for the pectoralis major?
Swimming?
Okay, so again, during that swim, and this I think would be more of a backstroke this time, right? You're going to bring the arm in, you're going to bring the arm up, and you got to immediately rotate it. So I guess maybe if we're coming across, I always think of this one in terms of like a steering wheel, right? If you got that steering wheel, right, and you need to make that sharp turn, you got to bring the arm up. As you're bringing the arm up, you need to immediately rotate it, and you need to bring it in towards the body. So you're kind of coming across this way.
Even like wiping a mirror?
Sure, there you go. Wipe it down. There you go. You going to demonstrate that for us?
Okay. So you would just spray and then wipe.
There you go. Yeah, not so much a circle, but absolutely that coming down, bringing it down across. Absolutely. That'd be a great example of that. Perfect. Excellent. All righty. So there you go. Now, I believe that only leaves us with one. How are we doing on time? That only leaves us with one more muscle that we need to know the origin and the insertion for. Oops, wrong button. There. Perfect. And that is the primary posterior muscle of the arm. The good news is we only have one more muscle to do, and then we'll call it a day because we'll have had all the origins and insertions in our pocket. The bad news is it's the tricep brachia. Firm slap. Yeah, firm slap would work too. Absolutely, right? Because you would you have to bring your arm up, you have to bring your arm in, bring it across. Yeah, I like that firm slap too. That'd be another good example. All right. Excellent. So one muscle, but it's the triceps brachia, the three-headed muscle in the posterior part of the brachial region. So we have 3 bellies we need to individually identify. So on the test, I can have 3 tags. Tag 1, tag 2, oops, tab 2, and tag 3. And for these 3 tags, I can ask the question, identify the muscle, be specific, or identify the origin, be specific. And for all 3 of these, those answers are different. However, for all 3 of these, if I ask insertion, their answer is the same. If I ask action, their answers are the same. So again, when you see that magic term, be specific. Oops, and now I need to not have it be white. You know I'm going to be talking about either an origin or an insertion, pardon me, an origin or name when I say that, and you need to be precise, okay? All right, now, as you can see here in the pretty picture, there are indeed 3 of them. The way I remember them, and this is what's easiest for me, is I find out here the lateral head is, of course, lateral, so that obviously makes it easy. But the lateral head is also over here lateral by itself, so the lateral head is lonely. Medial head, of course, is going to be medial, but notice the medial head is also the smallest of the 3 heads, so the medial is mini. And then, of course, that leaves us with the long head. And of course, as the long head indicates, the long head is long. In fact, it is so long, it leaves the brachial region and actually attaches to the scapula. So we have the lonely lateral, the mini medial, and the, well, long. They can be a little tricky to find, but the key is to find their tendon. The one nice thing about all of these is they all connect to the same common tendon, which is what attaches it to the back of the arm. I haven't bothered to look at the pictures from our model, but here we see it. Excellent. We can see this nicely here on our model. Well, okay, not nicely, but we can see some of it here. And again, the key of what you're going to be looking for is this nice big white tendon. This nice big white tendon is what all of the muscles of the tricep connect to. Notice over here, we have the lateral lonely by itself. And notice, like we saw in our bodybuilder, there are basically 3, when that bodybuilder gets really big, there are 3 muscle bellies you can see on the outside of their arm. The long head of the bicep brachia, the lateral head of the tricep brachia, and that brachialis in between. So if you remember that one bodybuilder picture, you could see those 3 bellies on his brachial region, and that is them there. Excellent. Notice here, we see the long head, which again is obvious because it leaves the humerus and goes up to the scapula. We can't fully see where on the scapula it is on the model, but it's there. And notice with the arm not fully rotated, we're looking more at a lateral view, we can only see a teeny bit of that mini medial one that is most distal and smallest of all of those. So here we see the 3 individual bellies on the model. We've seen the 3 individual bellies on the illustration. So let's draw the origins of the 3 individual muscles. Let's start first with the origin of the lateral head. What is the origin of the lateral head of the tricep brachia?
Posterior proximal humerus.
Excellent. This is a posterior muscle, so not surprisingly, it is on the posterior side. That lateral that is lonely is longer, so it comes further up the humerus, and so we attach to the proximal, lateral, and posterior part of the humerus. So it is proximal, it is of course posterior, and it is of course lateral, because it's the lateral head. What is the origin of the medial head? Oops, no. Shut. All right, hold on.
Posterior distal humerus.
There you go. So again, this is lateral. Still pink. It is lateral, it is proximal, and it is posterior. Excellent. So there we go. Now the medial head is small, so it's distal, it stays down here. It is, of course, posterior, and being the medial head, it is going to be medial. So if we think of its origin is going to be medial, as the name indicates, distal, because it is shorter, but it's still posterior, because it is part of the triceps brachia. So both are on the humerus. One's lateral, proximal, and posterior. The other's medial, distal, and posterior, but they're both on the humerus. And of course, the long head is so long, it leaves the humerus and goes to the scapula. And where is it on the scapula? Where does it originate? Excellent. On the infraglenoid tubercle. That roughened bump right here, that is its origin. Notice, and I'll color it in green so we don't get confused, but this view actually does a pretty decent job of showing us the supraglenoid tubercle, which remembers where the long head of the bicep brachia connects. So notice the long head of the bicep brachia connects to the supraglenoid tubercle. And the long head of the tricep brachia connects to the infraglenoid tubercle. So notice our two long heads of our two bicep and tricep brachial muscles connect to the superior and inferior glenoid tubercles above and below the scapula. I wouldn't say it's necessarily the strongest. It is the only one that crosses the shoulder, so it is the primary one involved in extending the elbow that way, so it might be a little bit stronger that way, although probably the efficiency of the lateral might be slightly bigger. But like I said, the good news is while we need to know their names, while we need to know their origins, we don't need to talk about their isolated actions. So we talk about their actions collectively. All right. So questions on the origins. The origins are the hard one. If we're comfortable with that, then we're home free. What is the insertion of the tricep brachia?
Common tendon into olecranon process of ulna.
There you go. Our olecranon process, right? Basically the point of our elbow, as we've talked about, right there is where it inserts. Notice then two things. It is going to, thanks to the long head, it crosses the shoulder and it crosses the elbow, so it crosses two joints. And it is on the ulna, the stable bone. So the tricep brachia cannot pronate or supinate. So it crosses two joints. It is a posterior muscle. How many actions does it have?
One.
Well, it has two joints, so it has to at least have, if it crosses two joints, it has to at least affect each joint once. So it indeed has two actions. And what are those two actions? What effect does a posterior muscle have on the shoulder? Extends the shoulder. What action does a posterior muscle have on the elbow? Extends the elbow. Posterior muscles extend. So this one extends the elbow, and then it extends the arm. All right. Everybody's even, so we get to start around new. So let's start with group one. Group one. Actually, no, hold on. Who went last? I think let's actually have group four. Group four, what'd you come up with for the tricep brachia? You in there, group four?
Sorry. I was trying to figure out my mute button. So let's see. It's to stiff arm, block the haters.
There you go, exactly. Especially behind you, absolutely. You have to extend the elbow and you have to extend the arm. So absolutely. So this one, as you were talking about, definitely can hyperextend the arm that way. Bring it back behind, anatomical position. Excellent, 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 excellent. So we didn't quite finish quite as early as I would like, but we did get through everything I wanted to cover. I wanted to cover obviously all our lecture stuff, and I wanted to get through all of the muscles of the arm that we need to know the origins and the insertions of. As you can see, there's still a fair number of other muscles on the arm we have to cover, but without having to do the origins insertions, we can just focus on their locations and their actions, and so that will make it go a little bit faster. So I think I have punished you enough this week. And again, it wasn't punishment. Again, I told you guys not to take this class. But I thank you guys for your hard work this week. I fully appreciate that this was a challenging week with a lot of material and a lot of time. And so I really want to thank you for the time and effort that you've put into this. I do appreciate it. And I hope you realize that I'm working as hard as you are to try to help you with this. So I hope together we can get people to the grades that they want ultimately in this class. But for the next couple of days, the onus switches back to you. Now you need to continue to master the material we've covered here. And I strongly, strongly, and strongly encourage you to start looking ahead as well so that you see and appreciate what we're going to be doing next week. So hopefully most of what we do next week will be mostly review. So please take some time to look at it because the test is on Thursday. All right. Any questions on that? All right, so the last thing, and I hate that I feel that I have to say this every class, but I feel that I do. Be healthy, be safe this weekend, take care, be smart. And again, listen to the medical experts. Do not listen to the politicians, right? If you break a bone in your arm, that is a medical issue and you go to a doctor to have it resolved. You don't go a politician with a broken arm. Well, what we're dealing here with COVID is a medical issue. Listen to the medical experts. Do not listen to the politicians, please. Make smart decisions, make safe decisions. Have a great weekend, and I will see you all on Monday. Take care and have a good weekend.
Thank you.
Thank you.